Within the vast universe of life, there are patterns both immense and minute, rhythms that echo in galaxies and in cells alike. Tonight we turn our attention not to the stars above, but to the stars within the tiny organelles that glow with the quiet energy of existence. Imagine for a moment that the human body is not simply one whole being, but a community made of countless living units, each a cell, each humming with a world of activity. Inside these cells, there are structures so small that even with the aid of powerful microscopes, they appear only as tiny specks. And yet, despite their size, these structures carry out some of the most important work of all. They are the mitochondria, often called the powerhouses of the cell, and they are always at work, even now, as you lie at rest. Every heartbeat, every slow breath, every calm thought relies on them. Without mitochondria, the energy of life would not flow. They are not only organelles, they are reminders that life thrives on hidden processes, on silent activity that continues even while we sleep. In the body of an adult human, there are trillions of cells, and within those cells, countless mitochondria, each one engaged in a task that sustains you without fanfare, without pause. It is in their work that the chemistry of life transforms into the feeling of life itself. As you begin to settle into the rhythm of listening, imagine a gentle landscape deep inside a single cell. The borders of the cell form like a quiet shoreline, enclosing a sea of liquid called cytoplasm. Floating within are the organelles, each a small island of purpose. Among them, the mitochondria drift and anchor themselves, elongated or rounded in shape, like small boats set upon calm waters. Each one is built with two membranes, an outer surface smooth and enclosing, and an inner surface folded into delicate ridges. These folds, called cristae, are not random. They are carefully arranged, increasing the area within, so that energy production can be more efficient. When viewed closely, the inside of a mitochondrion resembles a labyrinth of curves and chambers, as though nature itself had designed a space for both beauty and function. It is easy to forget how small they are. A mitochondrion is so tiny that tens of thousands could fit within the width of a single strand of hair. Yet numbers do not lessen their significance. They are abundant because the demand for energy is constant, and so the cell provides many copies. In a muscle cell, where energy is required in great amounts, there may be hundreds or even thousands of mitochondria, each one contributing to the effort of movement, contraction, and repair. In a quiet skin cell, there may be far fewer. The number is matched to the need, as though the cell knows how much power its tasks require and equips itself accordingly. In their stillness, mitochondria are never idle. They are the furnaces of living matter, taking in the small molecules that come from food, the sugars broken down from fruits and grains, the fatty acids released from oils and stores, and turning them into something more direct and more useful. This something is adenosine triphosphate, or ATP, often described as the currency of energy. Just as societies rely on currency to exchange value, cells rely on ATP to transfer energy. Each molecule of ATP carries a small packet of potential, ready to be spent whenever the cell needs to move, to build, to communicate. Without ATP, the cell would fall silent, unable to perform even the most basic tasks. And so mitochondria are not simply parts of the cell. They are engines, transforming what is available into what is essential. The process is continuous, reliable, and precise. It is easy to imagine the mitochondria as tiny glowing hearths, each one radiating quiet warmth, each one providing the spark that allows life to carry on. They do not clamor for attention. They do not boast of their labor. They work quietly, rhythmically, and without rest. In this way, they embody the very essence of life's persistence. Small, silent, ceaseless. As you listen, Allow yourself to picture the scene inside a cell, as though it were a world of its own. The cytoplasm becomes a gentle ocean, the organelles floating like islands, and the mitochondria glowing softly as lanterns beneath the surface. Each fold of the cristae is a chamber where the dance of electrons takes place, where chemistry becomes vitality. Imagine drifting closer, watching as nutrients arrive, as oxygen flows in, as invisible reactions unfold with a grace that belies their complexity. It is a quiet ballet of molecules, an unseen choreography that takes place billions of times each second across the expanse of your body. When we call the mitochondria the powerhouse of the cell, it is not a metaphor chosen lightly. They truly are the seat of energy conversion, and it is through them that the act of eating, breathing, and moving converges into life itself. Each bite of food, once digested and absorbed, offers up molecules that mitochondria can use. Each breath of air brings oxygen, which these organelles require to finish the process. Without mitochondria, the food would remain unspent fuel, the oxygen unused potential. Together, they make energy possible. They allow the story of life to continue. Think for a moment of your own breathing. As the chest rises and falls, oxygen enters the lungs, travels into the bloodstream, and reaches each cell. There, the oxygen finds its way into mitochondria, where it serves as the final step in the chain of reactions. Without it, the energy process would stall. With it, the mitochondria are able to release the energy stored in food and hand it over in the form of ATP. This deep connection between breathing and cellular power is one of the most intimate relationships in biology. You inhale, not only for yourself, but for the quiet engines within you. Even now, as you rest, the mitochondria are at work in every part of your body. In the brain, they provide the energy that allows neurons to send signals, weaving thought, memory, and dreams. In the muscles, they generate the strength that keeps your heart pulsing and your lungs expanding. In every organ, every tissue, every cell, they provide the subtle energy that maintains balance, warmth, and life. It is comforting to remember that this process is continuous. There is no need for command or conscious effort. The mitochondria work whether you are awake or asleep, whether you are moving or still. They are the quiet workers of life, present from your earliest days as an embryo, persisting through growth, change, and age. They link you to all living beings, for every animal, every plant, every fungus shares in their presence. They are universal, ancient, and indispensable. As the mind drifts further into rest, think of the mitochondria as soft lights glowing within you, a constellation of inner stars. They flicker not with fire, but with chemistry, each spark an act of transformation, each act a gift of life. They ask for nothing, but provide everything. Their hum is steady, a rhythm without sound, a music without note, and it is in their silence that life finds its strength. And so the story begins. With the mitochondria, we enter the world within, a place of structure, of balance, and of quiet power. This is the soft opening to a journey that will take us through their form, their function, their history, and their mysteries. But for now, it is enough to know that they are with you, working ceaselessly, faithfully, keeping the light of life glowing gently within. 2. The cell as a landscape. When we shift our gaze from the level of the whole body to the intimate scale of the cell, it is as though we are entering an entirely new world. Each cell can be imagined as a self-contained landscape, a small realm enclosed by delicate walls, filled with movement and subtle order. The cell is not a featureless space, nor is it a chaotic soup. Instead, it is a place where countless structures coexist, each with its purpose, each woven into the greater pattern of life. In this landscape, Mitochondria play their crucial role, but to truly understand their significance, we must first appreciate the surroundings in which they reside. Picture, if you will, the cell as a sphere of quiet boundaries, its membrane like a thin but protective shoreline, separating the inside world from the external environment. Beyond that border lies the body's tissues, its blood, its flowing streams of nutrients and signals. Within the cell's walls, a semi-fluid substance called cytoplasm forms the terrain, soft and yielding, allowing structures to float as though suspended in a gentle sea. This is the stage upon which all organelles perform their timeless work. Within this cytoplasmic sea are the organelles, each like an island or a landmark. Some are large and imposing, such as the nucleus, which holds the DNA, and thus the instructions for life itself. Others are small and numerous, like ribosomes, scattered across surfaces or drifting freely, translating genetic codes into proteins that build and repair. The endoplasmic reticulum stretches like folded hills, connected to the nucleus, its membranes providing a surface where proteins and lipids are crafted. The Golgi apparatus, stacked like flattened stones, receives these creations and refines them, packaging and preparing them for use or for transport out of the cell. Lysosomes patrol quietly, holding enzymes that can break down waste, recycling the parts into usable form. Each structure, though distinct, is part of the harmony of the cell. And then, scattered among them, are the mitochondria. They do not dominate in size, yet they are ever present, sprinkled throughout the cytoplasm, sometimes moving, sometimes remaining in place. Their shape varies. Some appear elongated, others rounded. In some cells, they drift in chains or clusters, while in others they are distributed more evenly. They respond to the needs of the cell, gathering where energy demand is greatest. In a nerve cell, they may cluster near synapses, ensuring that the energy required for signaling is always available. In a muscle fiber, they align along the contractile elements, supplying power directly to the machinery of movement. This is why it is helpful to think of the cell as a landscape. Every feature exists in relation to the others, every organelle performing its role while depending on the presence of the rest. If mitochondria are the furnaces, then the nucleus is the library, the ribosomes are the builders, and the Golgi the distributor. The cytoplasm is the field upon which all activity unfolds. It is not that one structure alone defines the cell, but rather the way all of them cooperate. In this community, mitochondria are essential, converting raw materials into the energy that fuels every other task. Without them, the builders cannot build. The library cannot be read. The membranes cannot maintain their balance. The cell, therefore, is not simply a container. It is a miniature ecosystem with flows of matter and energy, with balance maintained through constant exchange. Nutrients arrive, signals are received, waste is removed, proteins are made, lipids shaped, enzymes activated, and through it all, mitochondria supply the currency that allows these processes to occur. This energy, ATP, circulates like a resource, spent in countless ways, always replenished by the steady work of the mitochondria. If we pause to reflect on this, it is striking how ordered the cell truly island. Though it contains thousands upon thousands of molecules in motion, it does not collapse into confusion. Instead, the molecules follow paths, guided by structure, by chemistry, by the invisible laws of physics that direct their interactions. Organelles provide compartments, separating processes so they can unfold efficiently. This compartmentalization is one of the great features of eukaryotic life, and mitochondria, with their double membranes and internal folds, embody this principle beautifully. Their inner chambers allow for specialized chemistry, while their outer boundaries keep those processes contained. And yet, The landscape is not static. Organelles are not fixed in place like monuments. They move, shift, and adapt. Mitochondria, in particular, are dynamic. They can fuse together, forming larger units when needed, or split apart, creating more numerous smaller ones. This balance of fusion and fission allows the cell to respond to changing demands, to stress, to growth. They are more like living colonies than static machines, adapting to ensure that energy production is never interrupted. In this way, the landscape of the cell is alive with rhythm. Imagine watching from within, observing mitochondria slowly migrating through the cytoplasm, drawn to where energy is required. Ribosomes chatter away on strands of messenger RNA, translating the genetic script into chains of amino acids. Proteins fold, membranes shift, vesicles bud and travel. It is a quiet but ceaseless activity, as though the cell is a city that never sleeps. For those who study biology, it is often tempting to divide these structures, to label them separately and describe their roles as if they were independent. But in truth, the cell operates more like an orchestra. Each organelle is an instrument, and though one may carry the melody at times, the harmony is only possible when all play together. The mitochondria provide the energy, but they rely on proteins encoded in the DNA of the nucleus and made by ribosomes. The ribosomes require ATP to function. The Golgi apparatus packages proteins, many of which will be destined for the mitochondria themselves. And so it continues, a cycle of mutual dependence. This interconnection mirrors larger patterns in nature. Just as an ecosystem on Earth requires plants, animals, microbes, and climate to work together, so too does the cell require its organelles. And just as ecosystems depend on flows of energy, so too does the cell rely on the mitochondria. They are the quiet centers, turning the wheel, sustaining the current. As we picture this inner landscape, we may find it comforting to imagine it as calm and balanced. Even though molecules collide and reactions occur at astonishing speed, the impression from a distance is one of harmony. Each organelle has its place, each process its rhythm, and the mitochondria form the heartbeat of this hidden world. They are not alone, but they are central, woven into the very fabric of cellular life. And so, the cell reveals itself not as a single, simple entity, but as a realm filled with variety, coordination, and quiet beauty. Within this world, mitochondria take their place, not as solitary workers, but as members of a greater whole, contributing their power to the community of organelles. They are both distinct and connected, both independent in structure and integrated in purpose. To see the cell in this way is to see life itself as a tapestry, each thread essential, each pattern dependent on the weave. As the mind drifts gently toward rest, the cell may be pictured as a luminous globe, glowing softly in the vastness of the unseen. Within, the organelles shimmer, and the mitochondria hum, sending out energy like ripples in a still pond. It is a landscape of balance, of flow, of quiet cooperation. And as you imagine this inner world, you are reminded that it exists within you, countless times over, sustaining you, shaping you, and keeping the light of life glowing gently, endlessly, within. 3. The shape of mitochondria. When we imagine the mitochondria, it is easy to think of them as simple ovals or tiny beans, as they are often drawn in textbooks. Yet their shape, though subtle, holds great importance. The mitochondrion is not a plain sphere, nor is it a rigid structure. It is flexible, adaptable, and carefully designed by nature to carry out its role. If we were able to shrink ourselves down, to drift into the cell and float beside one, we would see that its form is at once modest and remarkable, a structure both efficient and elegant. The mitochondrion is enclosed by two membranes, like two layers of fabric wrapped around a core. The outer membrane is smooth, almost unremarkable at first glance, serving as the boundary that separates the organelle from the rest of the cytoplasm. This outer layer is permeable to many small molecules, allowing nutrients and signals to pass through with relative ease. It functions as a gentle gatekeeper, a thin but steady wall that defines where the mitochondrion begins and where the cytoplasm ends. Inside lies the inner membrane, and it is here that the artistry of the mitochondrion becomes apparent. Unlike the smoothness of the outer layer, the inner membrane folds inward again and again, creating ridges known as cristae. These folds are not random. They are purposeful, designed to increase the surface area within the mitochondrion. Each curve, each fold, is an expansion of space, providing more room for the essential machinery that produces energy. If the inner membrane were stretched out flat, it would cover far more area than the organelle's size would suggest. In this way, nature has found a way to fit great capacity into a tiny volume. The cristae are more than simple ridges. They are like shelves lined with enzymes and proteins, each one involved in the transfer of electrons and the production of ATP. They are the true workspace of the mitochondrion, the place where the chemistry of energy unfolds. As electrons pass along chains of proteins embedded in the membrane, protons are pumped across, building a gradient, a tension of sorts, that drives the creation of ATP. All of this depends on the arrangement of the inner folds. Without them, there would not be enough room for the many complexes required. With them, the mitochondrion becomes a compact but powerful site of energy conversion. Beneath these folds lies the matrix, a gel-like substance that fills the innermost space. The matrix is not empty. It contains enzymes that play vital roles in breaking down molecules, in processing the products of food into forms that the inner membrane can use. It also contains the mitochondrial DNA, small circular strands that speak of ancient origins, as well as ribosomes and other machinery for producing proteins. The matrix, though less visually striking than the folds of the membrane, is the heart of the mitochondrion's inner life, the ground upon which many essential reactions take place. The double membrane structure of the mitochondrion is one of its most distinctive features, and it hints at the story of its evolutionary past. Long ago, mitochondria are believed to have been free-living bacteria that entered into a partnership with larger cells. Their two membranes are a record of this union, one from their own bacterial ancestor, and one from the host cell that engulfed them. To this day, their structure carries this memory, an echo of an event that shaped the course of life on Earth. Though often drawn as static ovals, mitochondria are not fixed in shape. They are dynamic, shifting, and changing as the cell requires. They can lengthen into threads or break apart into smaller units. They can merge with one another, fusing into larger structures, or divide, producing more mitochondria when the demand for energy rises. This ability to alter their form is not merely a curiosity. It is central to their function. A network of elongated mitochondria may be more efficient at supplying energy across a large cell, while smaller, separate mitochondria may allow for quicker responses in localized areas. In this way, their shape is never final but always adapted to the needs of the cell. This dynamism also allows mitochondria to move. Guided along the cytoskeleton, they travel within the cell to where they are needed most. If a region of the cell is engaged in heavy activity, mitochondria may drift closer, clustering around the site to provide energy directly. If another part of the cell falls into disuse, the mitochondria may drift away, conserving resources. This mobility, combined with their changing shape, makes them more than passive organelles. They are active participants, responding to the living needs of the cell moment by moment. From a distance, one might imagine mitochondria glowing softly, their folds like illuminated ridges, their matrix alive with chemical motion. Their outer membrane forms the simple outline, but the inner folds give them texture, depth, and purpose. They are like lanterns with intricate interiors, their structure perfectly suited to the flow of electrons, the transfer of energy, the birth of ATP. The importance of their shape can also be seen when it falters. If the inner folds become damaged, if the membranes lose their integrity, the mitochondrion cannot function as it should. Energy production slows, and the cell suffers. In this way, the structure of the mitochondrion is not merely a matter of form, but of survival. The folds are life-giving, the membranes protective, the shape itself a reflection of purpose. It is worth pausing here to reflect on the balance of beauty and utility in this design. The cristae, curling inward like delicate waves, are as pleasing to the eye as they are effective in their role. The double membrane, enclosing yet permeable, is simple and profound. The matrix, quiet and unassuming, holds the very instructions that tie the mitochondrion to its ancient past. Together, these elements create a form that is both functional and graceful. As you imagine this shape, let it become clear in your mind. A small capsule, smooth on the outside, folded within, alive with unseen activity. The folds are not empty lines, but busy streets, lined with workers, each passing electrons, pumping protons, building gradients, crafting ATP. The matrix is not a void, but a field, filled with enzymes and DNA, a storehouse of history and activity. The membranes are not barriers, but boundaries, defining and protecting, allowing just enough exchange to sustain the process. And remember that these shapes are not rare. They are countless, replicated across your cells, present in nearly every tissue of your body. Wherever there is energy to be made, mitochondria are there, their forms repeated in quiet abundance. Their shapes, though unseen, are constant companions, always working, always adapting, always sustaining life. As the mind settles into the rhythm of this thought, the mitochondria can be seen not merely as parts of biology, but as quiet sculptures, works of natural design, whose shape embodies their purpose. They are small, yet profound, ordinary in appearance, yet extraordinary in what they make possible. And as you drift deeper into rest, you carry with you the knowledge that within you, millions upon millions of these shapes continue their ceaseless work, folded and curved, keeping the gentle flame of life alight. For energy is life's currency. In the quiet interiors of every cell, a special kind of currency circulates, not coins of metal or paper notes, not the currencies of human trade, but a molecular token far older and more universal. This currency is adenosine triphosphate, more gently known as ATP, and it serves as the means by which energy is transferred, stored, and spent across all of life. The mitochondria, with their folded membranes and ancient machinery, are the primary mint of this currency, producing it in constant abundance so that cells can go about their endless work. Without ATP, the motions of life would falter. Muscles would not contract, neurons would not fire, proteins would not fold, and thoughts themselves would fade into silence. To understand the mitochondria is, in essence, to understand ATP, for it is this molecule that they create, this coin of energy that they place into the hands of every process within the cell. ATP is a small molecule, but its power lies in its bonds. It is composed of adenosine, a structure made of adenine and ribose, bound to 3 phosphate groups arranged in a line. The last 2 phosphate bonds are rich in potential energy, like springs held under tension. When one of these bonds is broken, the spring is released, and energy flows into the cell. The molecule becomes adenosine diphosphate, or ADP, now with 2 phosphates instead of 3. In that release, the currency has been spent, energy made available to power whatever process required it. Just as coins are passed from hand to hand, ATP is passed from mitochondria to enzymes, from enzymes to reactions, fueling countless activities in rapid succession. The beauty of this system is in its reversibility. Once ATP has been spent, once the third phosphate has been released, mitochondria can take ADP and attach a phosphate once more, recreating ATP. The coin is reminted, ready for use again. This cycle of spending and replenishing occurs continuously, endlessly, as long as life persists. In fact, the human body turns over an astonishing amount of ATP every day, producing and consuming its own weight in this molecule. At any given moment, only a small amount of ATP is present in the body, for it is used so quickly. The mitochondria must therefore work tirelessly to keep the supply constant, ensuring that no process is left unfunded. Imagine for a moment the cell as a city. The organelles are its districts, the proteins its workers, the membranes its walls. ATP flows through this city like coins in the marketplace, exchanged in every corner, required by every activity. A motor protein, pulling a vesicle across the cytoplasm, spends ATP with each step. An enzyme folding a chain of amino acids into a protein shape uses ATP to guide and correct. A muscle fiber contracting in your arm spends trillions of ATP molecules in mere seconds. The city never stops, and neither does the flow of this currency. Mitochondria are like the banks, the power stations, the mints, providing a steady stream of coins to keep the city alive. ATP is not only useful for powering processes but also for regulating them. The concentration of ATP within a cell acts as a signal, informing the machinery of how much energy is available. When ATP levels are high, the cell knows that energy is abundant, and it can proceed with tasks of growth and synthesis. When ATP levels fall, the cell senses scarcity, shifting into conservation, slowing certain processes until the balance is restored. In this way, ATP serves as both currency and measure, a token of value and a gauge of abundance. The mitochondria, by controlling the flow of ATP, in effect set the pace of life within the cell. This currency is universal across nearly all forms of life. From the smallest bacterium to the largest animal, ATP is the coin of choice. There is a deep poetry in this, for it means that every living being, no matter how different, relies on the same molecule to carry out its work. The firefly lighting the night sky, the whale diving into deep oceans, the oak tree stretching its leaves toward the sun, all are sustained by the same energy token. It is one of the great unifying features of biology, a reminder that beneath the variety of forms and behaviors, life shares common threads. The way ATP is used varies widely, but its principle remains the same. It releases energy by breaking a bond, and that energy is harnessed. In neurons, ATP helps maintain the delicate balance of ions across membranes, creating electrical signals that ripple through the brain and body. In glands, ATP drives the secretion of hormones, releasing chemical messengers into the bloodstream. In immune cells, ATP fuels the rapid responses needed to recognize and confront invaders. Even in the simplest tasks, such as maintaining the structure of the cell itself, ATP is required. The cytoskeleton, the network of fibers that gives a cell its shape, relies on ATP-powered proteins to assemble, disassemble, and shift as needed. Without ATP, the cytoskeleton would collapse, and the cell would lose its form. It is striking to realize how short-lived each ATP molecule is once it is created. Within seconds of being minted, it is usually spent, passed along to some waiting process. This means that mitochondria must be constantly active, producing ATP in immense numbers. They are like forges that never cool, furnaces that burn without smoke, producing energy not in bursts, but in steady streams. In a single muscle contraction, Millions upon millions of ATP molecules are consumed and replaced, their creation and destruction so rapid that it blurs into a continuous hum of activity. Because ATP is so central, cells maintain elaborate systems to ensure its supply is never interrupted. Mitochondria are the primary source, but they are not the only one. Some ATP is generated in the cytoplasm through glycolysis, a pathway that breaks down glucose into smaller pieces. Yet this method yields only a small amount of ATP. The mitochondria, with their inner membranes and electron transport chains, produce the overwhelming majority. They are the factories of energy, and without them, the city of the cell would quickly run out of currency, its work grinding to a halt. The idea of ATP as currency is not merely metaphorical but deeply functional. It is the unit of trade, the transferable token that allows reactions to occur that otherwise would not. Many chemical reactions in the cell require energy input, and on their own, they would never proceed. By coupling these reactions to the breakdown of ATP, the cell makes them possible. It is as though ATP pays the cost, offering its energy to balance the equation, allowing the reaction to unfold. In this way, ATP is not only a store of energy but also a universal facilitator, enabling life's chemistry to flow. As you drift further into thought, imagine ATP not as an abstract molecule but as a gentle pulse, a rhythm of creation and release, flowing through the cell like waves upon a shore. Each wave rises as mitochondria attach a phosphate, creating ATP, and each wave falls as the bond is broken. The rhythm is ceaseless, the pulse unbroken, carrying life forward in silence. It is a hum too soft for ears, too quick for eyes, but it is there, always, in every breath, every movement, every dream. And so, ATP remains the quiet coin of life, minted by the mitochondria, spent by the cell, replenished in endless cycles. Its value never changes, its role never diminishes. It is universal, indispensable, and timeless. The mitochondria, in their folded shapes, exist to produce it, to keep the currency flowing, to ensure that life's work never falters. As you rest, the forges continue, the coins are minted, and the currency circulates. It is the silent economy of life, an economy that never pauses, never depletes, as long as mitochondria endure. 5. From food to energy. Every living being relies on energy. And for us, this energy comes from the food we eat. A piece of fruit, a slice of bread, a sip of milk, each carries within it chemical bonds that hold potential energy, captured from the sun through plants, or gathered from other animals that in turn relied on plants. When we eat, we are part of this ancient chain, taking in molecules that our cells will carefully break down and transform into a usable form. The mitochondria stand at the center of this transformation, turning the varied foods we consume into the universal currency of ATP. Without this conversion, the energy stored in food would remain locked away, inaccessible to the countless processes that depend upon it. When food enters the body, digestion begins, and the complex molecules of proteins, carbohydrates, and fats are gradually broken apart. Proteins are reduced to amino acids, carbohydrates to simple sugars, such as glucose, and fats to fatty acids and glycerol. These smaller units are absorbed into the bloodstream and delivered to cells throughout the body. At this point, the nutrients are like raw materials piled at the city gates, ready to be used, but not yet in a form that can be spent. To be transformed into energy, they must enter the pathways of metabolism, and ultimately, they must reach the mitochondria. Take glucose, the simplest and most central of these fuels. Once inside the cell, glucose begins its journey through glycolysis, a series of reactions that occur in the cytoplasm. Glycolysis does not require oxygen, and it breaks glucose into two smaller molecules of pyruvate, producing a small amount of ATP and a few electrons carried by special molecules called NADH. This is a modest start, but not the main prize. The real energy lies beyond, in the mitochondria. Pyruvate molecules are transported into the mitochondrial matrix, where they are further processed. Each is converted into acetyl-CoA, a molecule that carries the fragments of glucose into the citric acid cycle, also known as the Krebs cycle. Within the citric acid cycle, acetyl-CoA is gradually dismantled, its carbons released as carbon dioxide, its electrons harvested and transferred to carriers such as NADH and FADH. These carriers are like shuttles, each holding onto high-energy electrons and preparing to deliver them to the inner membrane. The cycle itself produces only a small amount of ATP directly, but its true importance lies in filling the electron carriers. They are the messengers, bringing energy-rich electrons to the inner folds of the mitochondrion, where the electron transport chain resides. The electron transport chain is the grand stage of energy transformation. Here, electrons are passed from one protein complex to another, moving along the cristae of the inner membrane like a current flowing through wires. With each transfer, a small amount of energy is released, and this energy is used to pump protons across the membrane, from the matrix into the intermembrane space. Gradually, a gradient is built, with protons crowded on one side, eager to flow back through the membrane. This gradient is a kind of stored energy, a tension ready to be released. At the end of the chain, the electrons must find a resting place. Oxygen serves this role, accepting the electrons and combining with protons to form water. This is why oxygen is essential. It is the final electron acceptor, without which the chain would halt and ATP production would collapse. With oxygen present, however, the process continues smoothly, and the gradient across the inner membrane grows stronger. The protons, now concentrated in the intermembrane space, flow back into the matrix through a remarkable protein called ATP synthase. This enzyme acts like a turbine, rotating as the protons pass through, harnessing their movement to attach phosphate groups onto ADP, forming ATP. With each turn, new molecules of ATP are minted, ready to power the cell's many demands. This entire process, from the passing of electrons to the pumping of protons to the turning of ATP synthase, is known as oxidative phosphorylation, and it is the primary means by which mitochondria produce ATP. The efficiency of this system is striking. From a single molecule of glucose, the mitochondria can produce up to 30 or more molecules of ATP. Compared to the 2 ATP molecules gained from glycolysis alone, this is a dramatic increase, made possible only by the inner structure of the mitochondrion and the presence of oxygen. In this way, mitochondria transform a simple sugar into a wealth of energy, enough to sustain the complexity of human life. Fats follow a similar but slightly different path. Fatty acids are broken down through a process called beta oxidation, occurring within the mitochondrial matrix. Each cycle of beta oxidation shortens the fatty acid chain by 2 carbons, producing acetyl-CoA and electron carriers in the process. The acetyl-CoA then enters the citric acid cycle, while the carriers deliver electrons to the electron transport chain. Because fatty acids can be very long, this process yields enormous amounts of ATP, making fats a dense and efficient energy store. This is why the body relies on fat for long-term energy storage, and why mitochondria are so important in mobilizing these reserves when needed. Proteins, though not usually the body's first choice for energy, can also be fed into the mitochondria. Their amino acids are deaminated, stripped of nitrogen, and converted into intermediates that can enter the citric acid cycle. In times of scarcity, When carbohydrates and fats are insufficient, mitochondria can extract energy from proteins as well, ensuring that the cell never runs out of options. In this way, mitochondria serve as the central hub of metabolism, taking in diverse fuels and channeling them into a common pathway. Whether the starting point is glucose, fat, or protein, the endpoint is the same: the production of ATP through the coordinated processes of the citric acid cycle and oxidative phosphorylation. The mitochondria unify these inputs, turning the varied diet of life into a single, universal output. As you picture this process, it may help to imagine a great water wheel. The food molecules are streams flowing into the wheel, and as they turn it, energy is released. The water wheel then powers the machinery of the mill, grinding grain into flour, providing sustenance for the village. In the same way, mitochondria accept the streams of nutrients, channel them through their cycles, and use their energy to mint ATP, which then flows outward to sustain the entire cell. It is a cycle both simple and profound, a quiet transformation repeated countless times each second. Even now, as you listen, your mitochondria are carrying out these steps. Each breath you take brings oxygen, ensuring the electron transport chain can continue. Each meal you eat provides glucose, fatty acids, and amino acids, filling the citric acid cycle and supplying the carriers with electrons. The mitochondria knit these elements together, weaving them into the fabric of life. You need not direct them. The process is automatic, reliable, and ceaseless. From food to energy, from molecules to movement, the path is continuous. What begins as a bite of nourishment becomes the thought in your mind, the beat of your heart, the gentle rise and fall of your breath. The mitochondria stand at the center of this transformation, converting the potential of food into the reality of life. And as you drift toward rest, it is comforting to remember that they will continue their work, silent and tireless, ensuring that the energy of life never ceases to flow. 6. The flow of electrons. If we think of the mitochondrion as a workshop, then the electron transport chain is the central assembly line, where energy is carefully drawn out of molecules and converted into a usable form. This assembly line, however, does not involve gears or wheels or hammers striking metal. Instead, it is built upon the quiet movement of electrons, tiny particles of charge that drift and flow with remarkable precision. The electron transport chain is a masterpiece of nature's engineering, a system that takes high-energy electrons harvested from food and guides them step by step, releasing their energy in controlled increments, never in chaos, never all at once. In this way, the mitochondrion avoids waste and builds a reservoir of power that can be tapped to form ATP, the currency of life. Electrons themselves are not visible. They do not glow or spark as they move through the inner membrane of the mitochondrion, yet their presence is real and their flow is constant. The carriers NADH and FADH, filled in earlier steps of metabolism, arrive at the inner membrane like messengers bearing treasure. Each carries electrons gained from the breakdown of glucose, fats, and proteins. These carriers dock with protein complexes embedded in the folds of the inner membrane, delivering their electrons and beginning the process of transfer. The chain is composed of several large protein complexes, each with a distinct role. Complex I accepts electrons from NADH, while Complex II accepts them from FADH. These complexes pass electrons onward to a mobile carrier called ubiquinone, which moves through the membrane like a shuttle. From there, electrons are delivered to Complex III, then handed to another mobile carrier, cytochrome C, which brings them to Complex IV. At each step, the electrons lose a little energy, and this energy is used to pump protons across the inner membrane, from the matrix into the intermembrane space. The process is like climbing down a staircase. At every step, some energy is released, but rather than letting it vanish, the mitochondrion captures it to build a proton gradient. This proton gradient is the heart of the system. On one side of the membrane, protons accumulate in high numbers. On the other side, they are fewer. Nature dislikes imbalance, and so the protons long to flow back, to spread evenly across the space. Yet, they cannot simply pass through the membrane, for it is impermeable to them. The tension builds, like water behind a dam. This stored energy, known as the proton motive force, is the potential that will ultimately drive the production of ATP. At the end of the chain, the electrons must find rest. Oxygen, drawn into the body with each breath, arrives in the mitochondrion and accepts the electrons. With the addition of protons, oxygen becomes water, a quiet and harmless product of the process. This simple act of acceptance is essential. Without oxygen, the chain would halt, electrons would remain stuck, and the entire flow would cease. This is why oxygen is the final electron acceptor, the keystone of the system. Without it, the assembly line of energy shuts down, and the mitochondrion can no longer produce ATP in abundance. The controlled movement of electrons is a delicate balance. If electrons were allowed to leap all at once from carriers to oxygen, the result would be an uncontrolled release of energy, like a spark or even an explosion. Instead, the chain slows their descent, breaking it into steps, each step coupled to useful work. This gradual release allows the mitochondrion to extract nearly all of the energy stored in food, conserving it in the form of a proton gradient, rather than losing it as heat. It is a model of efficiency, a quiet unfolding of power. Once the gradient is established, the protons flow back into the matrix through ATP synthase, a remarkable molecular machine. This enzyme is often described as a turbine, for it truly rotates as protons pass through, turning mechanical motion into chemical bonds. With each rotation, ATP synthase attaches a phosphate group to ADP, creating ATP. The flow of electrons has thus been translated into the flow of protons, and the flow of protons into the creation of energy currency. The chain is complete, from food to electrons, from electrons to gradient, from gradient to ATP. The elegance of the electron transport chain lies not only in its chemistry, but in its universality. Across all eukaryotic life, from single-celled organisms to humans, the same principles apply. The same complexes pass electrons, the same oxygen accepts them, the same ATP synthase spins. It is one of the most conserved and fundamental processes in biology, a system so effective that evolution has preserved it with little change for over 1 billion years. If one were to imagine this flow, it might appear like a river moving through a landscape of terraces. At the top, the river holds great potential energy, rushing with force. As it descends through channels and steps, the energy is harnessed to turn wheels, to power mills, to irrigate fields. By the time the river reaches the bottom, its energy has been spent usefully, and it continues gently onward. In the mitochondrion, the electrons are that river, descending, step by step through the chain, their energy captured at every stage to build the proton gradient. The final resting place is oxygen, waiting at the bottom like a calm pool, ready to receive what remains. The importance of this process cannot be overstated. It is the source of nearly all ATP in aerobic organisms, the reason why oxygen is vital, the mechanism that sustains muscles, nerves, and every activity of life. Without the electron transport chain, life as we know it could not exist. The quiet flow of electrons, invisible and constant, is the very heartbeat of energy. There are moments when the system produces byproducts, when electrons escape the chain prematurely and react with oxygen to form reactive oxygen species. These molecules, though potentially damaging, are also signals, guiding cells to adapt, repair, or respond. Even here, the mitochondrion is not careless. It balances the risks, uses the signals, and maintains its function. The flow of electrons is not perfect, but it is remarkably stable, a system that has endured through time because it is both powerful and resilient. As you imagine this process, let it appear in your mind as a gentle cascade. Electrons move step by step. Protons rise on one side of the membrane. A gradient builds like pressure behind a gate. Then, through ATP synthase, the protons flow back, turning the wheel, forging new ATP with each rotation. The cycle continues, unbroken, day and night, in every cell of your body. You do not sense it directly, but every breath you take sustains it. Every heartbeat depends upon it. Every thought and dream is powered by it. And so, the flow of electrons becomes more than a scientific detail. It is the quiet river within, the invisible current that drives the machinery of life. It is not loud, not violent, not chaotic. It is steady, precise, and ceaseless. As you rest, it continues, electrons stepping down their chain, protons moving back through their gates, ATP forming again and again. The mitochondria keep the current alive, and through them, you remain alive, carried forward by the flow of energy that has sustained life for countless generations. 7. The role of oxygen. Oxygen is one of the most familiar substances in our daily lives. We breathe it in with every gentle inhale, and we exhale carbon dioxide in return. Yet behind this simple rhythm lies one of the deepest connections between the outside world and the inner workings of our cells. Oxygen is not merely the air that fills our lungs. It is the final link in the chain of energy, the essential partner that allows mitochondria to transform food into life's usable power. Without oxygen, the flow of electrons in the mitochondria halts, the gradient across the inner membrane collapses, and ATP production slows to a near stop. In this sense, oxygen is more than an element. It is the quiet receiver, the silent guest at the end of the chain, waiting to accept electrons and bring closure to the process of energy creation. When we inhale, oxygen molecules enter the lungs and pass into the bloodstream, carried by red blood cells that are specially designed for this task. Hemoglobin, the iron-rich protein in these cells, binds oxygen tightly yet reversibly, allowing it to be transported to tissues throughout the body. Once delivered, the oxygen diffuses into cells and eventually reaches the mitochondria. It is here, deep within the folds of the inner membrane, that oxygen plays its critical role. At the end of the electron transport chain, electrons that have traveled from food molecules through carriers and protein complexes finally need a resting place. Oxygen provides that destination. By accepting these electrons and combining with protons, oxygen is transformed into water, a harmless and vital byproduct. This role may seem passive at first glance, but in truth, it is essential. If oxygen were absent, the electron transport chain would stall. Electrons would have nowhere to go, and the entire system would become clogged. The proton gradient would dissipate, ATP synthase would fall silent, and the cell would struggle to sustain itself. This is why oxygen is often described as the final electron acceptor. It is the endpoint that keeps the chain moving, the quiet anchor that allows the system to flow. The dependence on oxygen has shaped life in profound ways. Early organisms on Earth existed without it, relying on less efficient means of producing energy. They could ferment sugars to gain a small amount of ATP, but the yield was low. When oxygen began to accumulate in the atmosphere, thanks to the photosynthetic activity of ancient cyanobacteria, a new possibility emerged. Cells that learned to use oxygen in their mitochondria gained access to far greater energy yields, allowing them to grow larger, more complex, and more diverse. The partnership between mitochondria and oxygen was a turning point in evolution, opening the door to the rich variety of eukaryotic life that now fills the planet. Oxygen's role in energy production also explains why breathing is so deeply tied to life. Every inhale provides fresh oxygen to maintain the electron transport chain, while every exhale removes carbon dioxide, the byproduct of the citric acid cycle and cellular respiration. The rhythm of breathing is thus synchronized with the rhythm of mitochondria, a cycle of exchange between the environment and the cell. To breathe deeply is to fuel the mitochondria, to give them what they need to continue their ceaseless work. Even as you rest, your breathing sustains this process, ensuring that every cell receives its share of oxygen, that every mitochondrion can continue its silent task. The relationship between oxygen and mitochondria is not only functional but also delicate. Too little oxygen, and cells are starved of energy. This condition, known as hypoxia, can occur at high altitudes, where the air is thin, or in medical conditions, where oxygen delivery is impaired. In such cases, the body adapts by producing more red blood cells, or by shifting temporarily to less efficient forms of energy production. Too much oxygen, on the other hand, can also be harmful, leading to the generation of reactive oxygen species that may damage cells. The balance must be maintained, and mitochondria are at the heart of that balance. Inside the mitochondria, oxygen acts not with force but with quiet patience. It does not drive the electrons forward. It simply waits at the end, ensuring that the chain can continue. This subtle role is often overlooked, yet it is fundamental. Without oxygen, life as we know it would be impossible. It is a reminder that some of the most important roles in nature are not those that take center stage, but those that hold everything together in the background. Consider, for a moment, the connection between the air around you and the cells within you. Each breath draws in oxygen that was once part of the atmosphere, perhaps produced by a tree or a blade of grass far away. That oxygen enters your lungs, travels through your blood, and reaches your cells. There, within the mitochondria, it accepts electrons, becomes water, and helps create ATP. In this way, the air and your body are linked in a continuous cycle, an exchange that ties you to the living world. The mitochondria, deep within, are the meeting place where outside becomes inside, where the atmosphere sustains the inner fire of life. This connection is not unique to humans. Every animal that breathes shares in it. Birds soaring through the sky, fish drawing oxygen from water through their gills, mammals resting in the shade all depend on mitochondria and oxygen working together. Even plants, which produce oxygen through photosynthesis, rely on mitochondria at night to consume oxygen and produce ATP. The cycle of oxygen in the biosphere is thus shared by all, linking species and ecosystems in a common dependence. It is a bond that crosses boundaries, uniting the vast diversity of life through a single, invisible thread. The role of oxygen is therefore both scientific and poetic. It is the invisible partner, the quiet participant that makes energy possible. It does not demand recognition, yet it is always there, waiting at the end of the chain, ensuring that the flow of electrons continues. It is taken in without thought, released without effort, yet it sustains every beat of the heart, every flicker of thought, every gentle breath. As you listen, allow yourself to feel the rhythm of your own breathing. Each inhale carries oxygen to your mitochondria. Each exhale releases carbon dioxide into the world. This exchange has been ongoing since your first breath and will continue without pause. It is steady, gentle, and enduring. And beneath it all, mitochondria continue their ceaseless work, guiding electrons to oxygen, producing ATP, keeping life alive. In the stillness of rest, the role of oxygen becomes even clearer. It is the silent link between the world outside and the energy within, the final step that allows mitochondria to transform food into power. It is not loud, not forceful, but essential. And as you breathe calmly, know that each molecule of oxygen is finding its way into your cells, into your mitochondria, into the very flow of electrons that sustains you. Oxygen is not simply the air you breathe. It is life's quiet partner, ever present, ever patient, ever vital. 8. The cycle of ATP production. The story of energy within the cell is a story of cycles, of creation and use, of breakdown and renewal. At the center of this story stands ATP, adenosine triphosphate, the molecule that has already appeared as life's currency. But what makes this molecule so remarkable is not only its usefulness in fueling cellular work, but also the way it can be constantly replenished. The cycle of ATP production is a dance of chemistry that never stops, a loop that ensures the energy demands of life are always met. To understand this cycle is to see the quiet resilience of biology, a system that creates balance between giving and receiving, between using and restoring, between expenditure and renewal. At its heart, ATP is a molecule of 3 phosphate groups joined to adenosine. The outermost phosphate bond is the most energetic, and when this bond is broken, energy is released. The molecule becomes ADP, adenosine diphosphate, with only 2 phosphate groups. If the cell continues to need energy, even ADP can lose another phosphate, becoming AM, adenosine monophosphate. Each time, energy is released, powering muscle contractions, fueling protein synthesis, driving the signals of neurons, and supporting countless other processes. Yet the story does not end with the breakdown. For the system to endure, without this recycling, the cell would quickly exhaust its supply, unable to sustain its work. The mitochondria provide the machinery for this recycling. In their inner membranes lies the enzyme ATP synthase, a molecular motor that uses the proton gradient created by the electron transport chain to restore ADP into ATP. Protons, concentrated in the intermembrane space, flow back into the matrix through ATP synthase, and with each passage, the enzyme rotates. This rotation is not metaphorical but real, a mechanical turning of subunits powered by the flow of protons. As it turns, ATP synthase captures ADP and an inorganic phosphate, joining them to form ATP once again. The molecule is thus reborn, ready to leave the mitochondria and power the cell once more. This cycle of breakdown and renewal is astonishingly rapid. In a single second, billions of ATP molecules are used and replaced within the body. On the scale of a day, an adult human turns over an amount of ATP equal to their own body weight. Yet at any given moment, the actual pool of ATP present is small. The cycle is efficient because it is constant, the mitochondria producing ATP as quickly as it is consumed. It is not storage but flow that sustains life, a river of molecules always in motion, always circulating between use and renewal. The citric acid cycle, or Krebs cycle, provides the substrates that feed into this system. As glucose, fats, and proteins are broken down, their fragments enter the cycle, producing NADH and FADH, which deliver electrons to the electron transport chain. The chain, in turn, pumps protons across the membrane, building the gradient that drives ATP synthase. The process is seamless, a series of linked steps where the output of one becomes the input of the next. Each link in the chain is necessary, each one contributing to the whole. Beyond the mitochondria, cells have smaller ways of producing ATP. Though these are less efficient, glycolysis, which takes place in the cytoplasm, generates ATP directly by breaking down glucose into pyruvate. This process does not require oxygen, and though it yields only 2 ATP molecules per glucose, it can provide quick bursts of energy when needed. In muscle cells, for example, glycolysis can supplement mitochondrial ATP production during intense activity. Yet for sustained energy, the mitochondria remain indispensable, their cycle of oxidative phosphorylation providing the vast majority of ATP. One of the most elegant features of the ATP cycle is its reversibility. ATP can be broken down to release energy, but it can also be reassembled as long as the cell has access to nutrients and oxygen. This ensures that energy is never truly lost but only transformed. The bonds of ATP hold energy temporarily, like a spring wound tightly, and when the spring is released, work is done. Then the spring is wound again, the phosphate reattached, and the cycle continues. This principle of reversible bonds is what makes ATP such a perfect currency, one that can be spent and restored countless times without losing its value. The cycle of ATP also reflects the broader cycles of life. Just as the earth has cycles of day and night, of seasons, of water flowing from sky to soil and back again, the cell maintains its own internal rhythms. ATP is part of this pattern, always being created, always being used, always returning to its starting form. The constancy of these cycles provides stability. It allows life to continue not as a series of sudden bursts, but as a steady hum, a background rhythm that never ceases. If we imagine this cycle, it might look like a wheel turning endlessly. On one side, ATP is broken down into ADP, releasing energy into the cell. On the other, ADP is lifted back up into ATP by the work of ATP synthase, restored and ready for use. The wheel turns with each breath, with each molecule of glucose consumed, with each electron passed down the chain. The turning never halts, not during waking hours, not during sleep, not even in the quiet stillness of rest. It is a wheel that carries life forward, unseen but constant. This ceaseless renewal is what gives cells their resilience. Even in moments of great energy demand, such as during exercise, the ATP cycle accelerates to meet the need. Mitochondria produce ATP at a faster rate. Protons flow more quickly through ATP synthase, and the wheel spins faster. In quieter times, such as during sleep, the demand is lower, but the cycle continues, slower yet steady, maintaining the functions that keep the body alive. In this adaptability lies the strength of the system. It can rise to meet peaks of activity and fall into calm rhythms, always keeping pace with the needs of life. The cycle also serves as a reminder of connection. The oxygen we breathe, the food we eat, the movement of electrons, the flow of protons, the rotation of ATP synthase, all converge in this cycle. The external world and the internal cell are bound together in a loop of energy, with ATP as the link. Each meal, each breath, feeds into the mitochondria, and from them, ATP flows outward, fueling the very act of living. As the mind drifts toward calm reflection, it is soothing to think of this cycle as a gentle pulse within you. Each second, countless ATP molecules are created and consumed, broken and rebuilt, spent and replenished. The mitochondria keep the cycle alive, their inner folds turning food and oxygen into energy tokens, their enzymes restoring ADP into ATP again and again. The cycle is eternal as long as life endures, a loop without beginning or end, a rhythm that underlies every movement, every thought, every dream. And so, the cycle of ATP production is not just a biochemical detail. It is the pulse of life itself, the endless renewal that sustains us. It is quiet, efficient, and ceaseless, a reminder that life is not built on permanence, but on constant flow. Each ATP molecule lives only for a moment before being spent, yet the cycle ensures that another always takes its place. The wheel never stops turning, and in its turning, life continues, steady and unbroken. 9. Variations across tissues. Though every cell that contains mitochondria relies on them for energy, not all cells require the same amount of power. Just as some cities glow brightly with constant activity while others remain quieter and slower, the tissues of the body differ in their energy needs. And because of this, the number, size, and arrangement of mitochondria vary across different types of cells. Some are packed with thousands upon thousands of these organelles, while others manage with only a handful. This variation is not random. It reflects the unique demands of each tissue, the way each part of the body lives its own rhythm while still remaining connected to the whole. Take muscle tissue as the first example. Muscles are among the most energy-hungry tissues of the body. Every movement, every contraction, whether it is the lifting of a hand, the beating of the heart, or the subtle motions that keep posture steady, depends on a steady stream of ATP. Muscle fibers are filled with contractile proteins that slide past one another to shorten the cell and produce force. And this sliding requires enormous amounts of energy. For this reason, muscle cells, particularly those in the heart and in endurance muscles, are densely packed with mitochondria. In heart cells, mitochondria may make up nearly half of the cell's volume. They cluster close to the contractile fibers, ensuring that energy is delivered directly where it is needed. This arrangement allows the heart to beat continuously, tirelessly, without pause from the moment life begins until it ends. Skeletal muscle, the tissue that allows voluntary movement, also relies heavily on mitochondria, though the distribution depends on the type of muscle fiber. Some fibers, designed for endurance, contain vast numbers of mitochondria, enabling them to sustain activity over long periods. Others, built for quick bursts of power, contain fewer mitochondria and rely more on anaerobic pathways like glycolysis. The balance between these fibers reflects the type of activity the muscle performs, and in turn, the energy strategy that supports it. In all cases, mitochondria are present, adapted to the task, ensuring that the energy matches the demand. Nervous tissue presents another striking example. Neurons, the long branching cells of the nervous system, communicate through electrical impulses that travel along their axons and release neurotransmitters at their synapses. This process requires extraordinary amounts of ATP, not for contraction but for maintaining ion gradients across membranes. At rest, neurons spend much of their energy pumping sodium and potassium ions to maintain the conditions needed for signals to fire. When an impulse travels down an axon, the energy demand increases further, and at the synapse, vesicles filled with neurotransmitters must be released, recycled, and refilled. To meet these demands, mitochondria cluster in large numbers near synapses, ensuring that ATP is always available at the point of communication. The placement is precise, as though the cell directs mitochondria to where energy is most urgently required. This distribution supports the speed and reliability of thought, memory, and sensation. The liver, too, demonstrates the importance of mitochondrial variation. As the central hub of metabolism, the liver processes nutrients, detoxifies substances, produces bile, and regulates blood sugar. Each of these functions requires energy, and liver cells contain abundant mitochondria to support them. These organelles are not only producing ATP but also participating directly in metabolic pathways, such as the breakdown of fatty acids and the processing of amino acids. The liver's mitochondria are versatile, shifting their activity depending on the state of the body, whether it has just eaten, whether it is fasting, whether it is under stress. In this way, The mitochondria in liver cells are not merely power producers, but regulators, adapting their output to the larger needs of the organism. Other tissues, however, live at a quieter pace and contain fewer mitochondria. Skin cells, for example, divide and renew themselves regularly, but their energy demands are modest compared to those of muscles or neurons. They contain enough mitochondria to maintain their function, but no more. Red blood cells take this principle further. They lack mitochondria entirely. These cells specialize in carrying oxygen, and by shedding their organelles, they make room for hemoglobin, maximizing their ability to transport gases. Instead of generating their own ATP through oxidative phosphorylation, red blood cells rely on glycolysis, a less efficient but sufficient method for their needs. This absence of mitochondria is not a weakness, but an adaptation, reflecting the specialized role of red blood cells in the body. Even within a single tissue, variation exists. Different cells of the same organ may contain different numbers of mitochondria depending on their tasks. In the kidney, for instance, cells that actively transport ions and maintain concentration gradients are filled with mitochondria, while neighboring cells with less demanding roles contain fewer. In the eye, photoreceptor cells require large amounts of energy to respond to light and maintain their sensitivity, and mitochondria cluster densely near the regions where this activity occurs. This variation also extends to changes over time. Cells can increase the number of mitochondria in response to training or stress. Endurance exercise, for example, stimulates muscle cells to produce more mitochondria, enhancing their capacity to generate ATP and resist fatigue. This adaptation, known as mitochondrial biogenesis, is one reason why regular training improves stamina. Conversely, in conditions where energy demands decrease, cells may reduce their mitochondrial numbers or activity. The system is dynamic, adjusting to the ebb and flow of life's requirements. The variations in mitochondria across tissues highlight not only the adaptability of these organelles, but also the elegance of biological design. No energy is wasted. Each cell has exactly the number of mitochondria it needs, positioned where they will be most effective, shaped by the demands of the tissue. In this way, mitochondria are both universal and particular universal in their presence across nearly all cells, particular in their adaptation to each cell's role. As you reflect on this, imagine the body as a great landscape of varied terrains. In some regions, the hills are dotted with countless glowing lanterns, representing mitochondria working in abundance. In others, the lights are fewer, scattered sparsely but still sufficient to illuminate the needs of the place. The heart is a city of ceaseless glow, bright with mitochondria, powering each contraction. The brain is a network of lights clustered at every point of connection, ensuring the flow of thought. The skin is a quieter countryside, with fewer lamps, each steady but modest. Together, the whole landscape is illuminated, each region lit in proportion to its needs. This balance, this variation, is part of the larger harmony of the body. It shows how energy is distributed wisely, how each cell receives what it requires, how mitochondria adapt to serve every corner of life. They are not identical in every tissue, but they are always present, always providing, always sustaining, and in this variation, they reveal their versatility, their ability to shape themselves to the rhythms of the body, to the unique patterns of each tissue. Even now, as you rest, your mitochondria continue this work, glowing more brightly in some tissues, more softly in others, but always steady, always alive. They are scattered across your body in countless numbers, yet each one is serving a precise role, a quiet adaptation to the needs of its home. And in this way, the variations across tissues form not a discord but a symphony, each note tuned to perfection, each part contributing to the whole. 10 ancient origins. To truly appreciate the mitochondria as they exist today, One must journey back into the depths of time, to an era when the Earth was young and life was still finding its place upon the planet. The story of mitochondria is not simply the story of an organelle within our cells. It is a story of ancient partnerships, of survival and cooperation, of a union so profound that it changed the course of evolution forever. The origins of mitochondria stretch back more than 1 billion years, to a time when the Earth was inhabited only by single-celled organisms, when the skies and seas were different, and when the foundations of complex life were only beginning to take shape. In those early days, the world was filled with prokaryotes, the simple cells that lacked nuclei or internal compartments. Among these were bacteria and archaea, small yet resilient, each surviving in its own way, some relying on fermentation, others on primitive forms of metabolism. Life was diverse, but it remained limited. Without efficient ways to harness energy, cells could not grow too large, nor could they develop the complexity that later organisms would achieve. The Earth itself was changing as well. Cyanobacteria, capable of photosynthesis, had begun to release oxygen into the atmosphere. What had once been a rare element was becoming more common, and the environment was slowly shifting from one dominated by anaerobic life to one in which oxygen played a central role. For many organisms, this change was dangerous. Oxygen, though useful, is also reactive, capable of damaging cells if not carefully controlled. To survive in this new world, life would need to adapt. It was in this environment that a remarkable event occurred, one that would alter the trajectory of life permanently. A larger host cell, itself likely an archaeon, engulfed a smaller bacterium. Normally, such an encounter might have ended with the bacterium digested as food, its nutrients broken down to feed the host. But in this case, something different happened. Instead of being destroyed, the bacterium remained alive within the host cell, and over time, a partnership formed. The bacterium was skilled at using oxygen to extract energy from nutrients, producing far more ATP than the host could generate on its own. The host cell, in turn, provided protection and a stable environment for the bacterium to thrive. What began as a chance encounter became a relationship of mutual benefit. This theory, known as the endosymbiotic theory, explains the origins of mitochondria. It suggests that mitochondria were once free-living bacteria, independent organisms capable of surviving on their own. Through symbiosis, they became part of a larger whole, eventually transforming into organelles. Over millions of years, the bond deepened. The bacterium transferred much of its DNA to the host cell's nucleus, losing many of the genes that once allowed it to live independently. It became dependent on the host for survival, and the host became dependent on it for energy. The two were no longer separate beings, but one combined entity, stronger and more capable than either could have been alone. Evidence for this ancient origin can still be seen today. Mitochondria retain their own DNA, arranged not in linear chromosomes like those of the nucleus, but in small circular loops, resembling the genomes of bacteria. They also contain their own ribosomes, which more closely resemble bacterial ribosomes than those of the eukaryotic cytoplasm. Mitochondria replicate independently within the cell, dividing in a manner reminiscent of bacterial fission. Their double membranes, too, hint at this origin, one belonging to the engulfed bacterium, the other derived from the host cell's membrane during the act of engulfment. These traces are fossils within us, living evidence of a union that shaped the history of life. The significance of this event cannot be overstated. With mitochondria providing efficient energy through oxidative phosphorylation, host cells could grow larger and more complex. They could support more DNA, more proteins, and more intricate internal structures. This newfound energy allowed the evolution of eukaryotic cells, the kind that make up plants, animals, fungi, and protists. Without mitochondria, complex multicellular life as we know it might never have arisen. The diversity of creatures that now inhabit Earth, from the smallest insect to the largest whale, owes its existence to this ancient partnership. It is humbling to realize that every cell in your body carries within it descendants of these ancient bacteria. Each mitochondrion is a reminder of this merger, a living echo of that first encounter more than 1 billion years ago. They are not foreign to us now. They are fully part of us. Yet in their structure and behavior, they still whisper of their past. They are both self-contained and dependent, both ancient and modern, both individual and collective. This story of origins also carries a quiet lesson about the nature of cooperation. Life did not advance through competition alone, but through partnership. The host cell and the bacterium found strength in unity, combining their abilities into something greater. This theme is repeated throughout evolution, where cooperation often leads to survival and success. The mitochondria within us are perhaps the most intimate example of this truth, for they are not simply allies beside us, but allies within us, inseparable and essential. As the mind reflects on this story, it may help to imagine the scene of early Earth, with its seas filled with microscopic life, its skies gradually filling with oxygen, its landscapes still forming. Among countless cells drifting in the water, one chance encounter occurred, a host cell engulfing a bacterium. From that moment, the history of life changed. The world of simple cells gave rise to complex beings, to ecosystems, to diversity beyond measure. The mitochondria within us are living relics of that turning point, a reminder that life's greatest leaps often come not from isolation, but from connection. Even now, as you breathe, as your mitochondria use oxygen to produce ATP, you are participating in the continuation of that ancient relationship. The partnership begun billions of years ago continues in every heartbeat, in every thought, in every breath. It is seamless now, so deeply integrated that we cannot imagine life without it. And yet, beneath the surface, the traces remain, telling a story of origins, of unity, and of transformation. The ancient origins of mitochondria remind us that the small can shape the great, that even the tiniest events can ripple through time to alter the course of life. Within your cells, the legacy of that moment endures, a quiet symbiosis that fuels your every motion. The mitochondria are not only powerhouses, they are storytellers, carrying within them the memory of the Earth's earliest days, the whisper of a union that gave rise to all that followed. 11. A partnership that shaped evolution. The story of mitochondria is not only about their ancient origins, but also about the profound consequences of their union with the host cell. This partnership was more than survival. It was transformation. It was the moment when life crossed a threshold, leaving behind the limits of simplicity and stepping into the realm of complexity. The joining of a bacterium with a larger host cell was not just an event. It was a turning point in evolution, one that shaped the path of every plant, every animal, every fungus, and indeed every human being who has ever lived. To appreciate the mitochondria fully, one must understand how this union changed the course of life, not in small ways, but in the grandest sense imaginable. Before the rise of mitochondria, life existed in its simplest form. Bacteria and archaea dominated the earth, small cells with limited internal structure, able to survive but not to expand into complexity. Their methods of producing energy, though sufficient for their size, yielded little compared to what mitochondria would later provide. These organisms could not afford to grow very large, for the more a cell expands, the more energy is required to sustain it. Their DNA was small, their proteins limited, their lifestyles restrained by the modest yields of fermentation and anaerobic metabolism. Life had persisted this way for billions of years, quietly abundant yet restrained in scope. The introduction of mitochondria changed everything. Suddenly, cells gained a way to produce energy at a scale far beyond what had been possible before. By harnessing oxygen, mitochondria could extract far more ATP from the same nutrients. A single glucose molecule, which yielded only a small handful of ATP without oxygen, could now be transformed into dozens of ATP molecules within mitochondria. This increase in efficiency meant that cells could support larger genomes, more proteins, more intricate internal systems. They could afford complexity, and complexity became the path forward. The result was the rise of eukaryotes, cells with nuclei, with organelles, with internal compartments that allowed for specialization and coordination. The nucleus provided a secure home for DNA, protecting the genetic instructions while also allowing them to expand in size and sophistication. Other organelles developed, each with its task: the endoplasmic reticulum for building proteins and lipids, the Golgi apparatus for refining and shipping, lysosomes for recycling. All of this was made possible because mitochondria provided the energy to sustain it. Without their contribution, these compartments might never have evolved, for there would not have been enough energy to maintain them. The partnership between mitochondria and the host cell thus laid the foundation for multicellular life. Once single cells became capable of greater energy output, they could cooperate, joining together to form tissues and organisms. Cells could specialize, some focusing on contraction, others on communication, others on protection or support. This specialization allowed for the development of bodies with organs and systems, capable of movement, thought, and adaptation. From the simple act of one cell hosting another, entire worlds of life unfolded: forests, oceans filled with creatures, ecosystems of dazzling diversity. It is striking to consider how different life might have been without this partnership. If mitochondria had never joined with their hosts, the Earth might still be ruled by bacteria alone. The planet would be covered with simple life forms. But the complexity we see today, animals walking, birds flying, humans thinking, would not exist. The symbiosis that created mitochondria was therefore one of the most important events in the history of Earth. It was a moment when chance and necessity came together to produce a result that reshaped the biosphere. This partnership also speaks to the theme of cooperation in evolution. While much attention is given to competition, to the survival of the fittest, the mitochondria remind us that collaboration can be equally powerful. The host cell and the engulfed bacterium did not destroy one another. They found a way to live together. Each brought something the other lacked. The bacterium brought efficiency and energy production. The host brought protection and resources. Together, they achieved what neither could alone. It is a story of unity, of strength through partnership, and it is a story that continues within every living eukaryotic cell. The evidence of this partnership is still visible today. Mitochondrial DNA, though greatly reduced from its ancient form, remains separate from nuclear DNA, a small reminder of their once independent life. The proteins of the mitochondria are partly encoded by their own DNA and partly by the DNA of the nucleus, showing the deep interdependence that has developed. Neither could now function without the other. The host and the mitochondrion are fully bound, their partnership so complete that they can no longer be separated. They are one organism, one identity, yet within them lies the memory of two. This shaping of evolution did not occur overnight. It was the work of millions of years, of gradual adaptation, of countless generations refining the relationship. At first, the partnership may have been fragile, with conflicts to resolve and balances to be found. But over time, the union stabilized, and the result was a new kind of life, one capable of greater feats than either ancestor alone. Evolution is often slow, yet its results can be profound, and the mitochondria stand as one of the clearest examples of this truth. As one reflects on this, it is possible to see life itself in a new light. We are not simply single beings, but collectives, born from ancient partnerships. The mitochondria within us are not outsiders. They are family, passed down through generations, carried in the maternal line, present in every cell. They shape not only our biology, but also our history. For without them, our species would never have arisen. The laughter of children, the songs of birds, the blooming of flowers, all of these are echoes of that first partnership, of the day when a host cell and a bacterium chose not to part, but to remain together. The partnership that shaped evolution is, in the end, a story of connection. It tells us that life advances not only through struggle, but through unity, that the bonds we form can create possibilities greater than we imagine. Within your cells, mitochondria continue to honor that bond, working tirelessly to provide the energy that sustains you. They are your ancient allies, your silent companions, your constant partners, and as you rest, you carry within you the legacy of their union, a legacy that shaped the evolution of life itself and continues to power the journey forward. 12. Their own genetic identity. Among all the organelles within the cell, mitochondria hold a distinction that sets them apart. They carry with them their own genetic identity, a small but remarkable remnant of their ancient past. Unlike the nucleus, which houses the vast majority of the cell's genetic material in long strands of DNA, mitochondria retain their own DNA, arranged in tiny circular loops more reminiscent of bacterial genomes than of eukaryotic chromosomes. This presence of their own DNA is a living memory of their origins as free-living bacteria, a fragment of independence that remains within them, even as they function as part of a larger whole. Mitochondrial DNA, often abbreviated as mtDNA, is small compared to the vast expanse of nuclear DNA. In humans, it contains only 37 genes, a tiny fraction of the tens of thousands housed in the nucleus. Yet these genes are critical. They encode essential components of the machinery that produces ATP, including parts of the proteins that make up the electron transport chain and ATP synthase. Without these genes, mitochondria could not function. However, they are not entirely independent. Over the course of evolution, much of the original bacterial genome was transferred to the host cell's nucleus, leaving only this small remnant within the mitochondria themselves. In this way, the partnership deepened, with mitochondria and the nucleus becoming interdependent, each relying on the other to provide the full set of instructions. The presence of their own DNA also means that mitochondria can replicate independently of the cell's nucleus. They divide in a process that resembles the fission of bacteria, elongating and splitting into two. When a cell divides, its mitochondria are distributed into the daughter cells, ensuring that each inherits these essential organelles. The ability of mitochondria to replicate within the cell allows them to respond to demand. If energy needs rise, mitochondria can multiply, increasing their numbers to supply more ATP. Their genetic identity, though small, gives them the autonomy to participate actively in the life of the cell. Another remarkable feature of mitochondrial DNA is the way it is inherited. Unlike nuclear DNA, which comes from both parents, mtDNA is passed almost exclusively through the maternal line. This is because the mitochondria present in the egg are transmitted to the embryo, while the mitochondria in sperm are typically destroyed after fertilization. As a result, each person carries mitochondrial DNA that is a direct inheritance from their mother and from their mother's mother, stretching back, unbroken through the generations. This maternal inheritance has allowed scientists to trace lineages far into the past, constructing a genetic map of human ancestry that connects us to ancient populations and migrations. The concept of a mitochondrial Eve, the most recent common matrilineal ancestor of all living humans, arises from this unique inheritance pattern, reminding us that within our mitochondria lie the stories of our shared origins. Mitochondrial DNA is also more vulnerable than nuclear DNA. It lacks the protective histone proteins that shield nuclear DNA, and it resides close to the electron transport chain, where reactive oxygen species are sometimes generated as byproducts. This exposure means that mtDNA accumulates mutations more quickly than nuclear DNA. Some of these mutations are harmless, while others can impair mitochondrial function. In humans, defects in mtDNA are linked to a variety of diseases, often affecting tissues with high energy demands, such as muscles and the nervous system. These conditions highlight the essential role of mitochondria and the delicate balance required to maintain their genetic integrity. Despite its small size, mitochondrial DNA has been an invaluable tool for science. By comparing variations in mtDNA among individuals and populations, researchers have been able to reconstruct patterns of human migration, tracing how our species spread from Africa to every corner of the globe. MoutDNA has also provided insights into evolutionary relationships between species, confirming the common ancestry of eukaryotes and illuminating the pathways of evolution. In this way, the tiny loops of DNA within mitochondria serve not only the cell, but also the broader pursuit of knowledge, carrying within them clues to the history of life itself. The independence of mitochondria, though partial, gives them a unique character within the cell. They are both part of the whole and distinct, both organelles and remnants of ancient organisms. Their genetic identity ties them to their bacterial ancestors, while also anchoring them within the intricate system of the eukaryotic cell. They are, in a sense, storytellers, preserving within their DNA a fragment of the tale of life's great transitions. If one pauses to reflect, it is humbling to consider that within every cell of your body, there are echoes of ancient genomes, still active, still necessary, still participating in the creation of energy. These genomes are not as vast as those in the nucleus, but they are essential, a reminder that even the smallest instructions can carry immense importance. The mitochondria, through their DNA, connect us not only to our mothers and grandmothers, but to the very dawn of eukaryotic life, to the moment when a partnership between two cells reshaped the future. It is also worth contemplating the symmetry of this inheritance. Just as mitochondria are inherited only from mothers, so too do they sustain the life of every child, passing from one generation to the next as silent companions. They are not chosen, not altered by will, but handed down with continuity, carrying forward the lineage of life. Each mitochondrion in your cells contains DNA that links you to a chain of ancestors, stretching back unbroken through time. Within this chain lies the story of humanity, a genetic thread connecting us all. As you think of this, you may imagine mitochondria not only as powerhouses, but as small libraries, each containing a handful of precious scrolls, carefully copied and carried forward through time. These scrolls are not vast encyclopedias, but brief instructions, concise and direct, yet absolutely essential. Without them, the cell's machinery would falter. With them, life continues. Each mitochondrion carries these scrolls within its matrix, preserving them as faithfully as it preserves energy. And so, the mitochondria stand apart among organelles, distinguished by their own genetic identity. They are living evidence of the past, active participants in the present, and carriers of stories into the future. They remind us that within the vastness of the cell, individuality can persist, that even in the midst of cooperation, fragments of independence remain. Their DNA is a small circle, but its meaning is immense. It is the link between ancient bacteria and modern humans, between mothers and children, between evolution and memory. As you drift into calm reflection, it is soothing to remember that within you are countless mitochondria, each with its own identity, each carrying the same small circle of DNA, each working quietly to sustain your life. They are your companions, your inheritors, your partners in existence. Their genetic identity is not separate from yours, but part of it, woven into the fabric of your being, passed down through generations, enduring, unbroken, eternal. 13. Mitochondria as communicators. When we think of mitochondria, we often picture them simply as powerhouses, tirelessly producing ATP and providing energy for the cell. Yet their role extends far beyond energy. Mitochondria are not silent servants working in isolation. They are communicators, sending and receiving signals that influence the fate of the cell. They converse with the nucleus, with other organelles, and even with neighboring cells, shaping the rhythms of growth, repair, and survival. Their voices are not spoken in words but in molecules, in cascades of chemical signals that ripple across the cell like waves. To understand mitochondria fully is to see them not only as engines but also as participants in an ongoing dialogue, essential members of the cellular community, whose messages carry profound weight. One of the most important ways mitochondria communicate is through the regulation of apoptosis, the programmed death of cells. Apoptosis is not a sign of failure, but a necessary part of life. It allows the body to remove damaged or unnecessary cells, to sculpt tissues during development, and to maintain balance in adulthood. When a cell must be removed, mitochondria play a decisive role. They release proteins from the space between their inner and outer membranes, such as cytochrome C, which trigger a cascade of events leading to cell death. In this way, mitochondria act as guardians of integrity, deciding when a cell has reached the end of its useful life. This decision is not made lightly. It is carefully regulated, for unnecessary cell death could harm the organism. Yet when damage is beyond repair, mitochondria provide the signal, ensuring that the greater system is protected. This role highlights a paradox. The same organelles that sustain life through energy production also hold the power to bring it to an end. They are both creators and destroyers, balancing between vitality and termination. This balance is essential to health. Without apoptosis, damaged cells might linger, accumulating mutations and potentially leading to disease. With too much apoptosis, tissues might wither away. Mitochondria, through their signaling, maintain this delicate equilibrium, demonstrating that their communication is as important as their production of ATP. Beyond apoptosis, mitochondria communicate by regulating metabolism. They send signals to the nucleus about the cell's energy state, informing it whether conditions favor growth, repair, or conservation. These signals often come in the form of molecules such as ATP, ADP, NADH, and reactive oxygen species. When energy is abundant, mitochondria signal that the cell can proceed with biosynthesis, building proteins and lipids, dividing and expanding. When energy is scarce, mitochondria warn the cell to conserve, slowing growth and focusing on survival. In this way, mitochondria set the pace of life, their communication guiding the cell's decisions moment by moment. Reactive oxygen species, often thought of only as harmful, also serve as important signals, produced as byproducts of the electron transport chain. These molecules can activate pathways that strengthen the cell's defenses, enhance repair mechanisms, or adapt metabolism. In moderation, they are not poisons but messengers, warning signals that prepare the cell for stress. Only when their levels rise too high do they cause damage. Thus, even in their imperfections, mitochondria provide valuable communication, reminding the cell of the balance it must maintain. Mitochondria also converse with other organelles. Their relationship with the endoplasmic reticulum, for example, is close and constant. At points where their membranes come into contact, they exchange calcium ions, which act as universal signals in the cell. Calcium released from mitochondria can trigger muscle contraction, stimulate enzyme activity, or activate signaling pathways. By regulating calcium, mitochondria influence processes far beyond themselves, extending their communication into nearly every aspect of cellular function. The flow of calcium is like a language, and mitochondria are fluent speakers, shaping the conversation with precision. Their communication extends outward as well, influencing neighboring cells and even the whole organism. When tissues experience stress, mitochondria release signals that can attract immune cells, prompting repair or defense. In times of energy, abundance, or scarcity, they help regulate hormones and metabolic pathways that affect the entire body. Mitochondria are thus not only participants in the life of a single cell, but contributors to the broader symphony of the organism. Their signals ripple outward, ensuring that tissues work in harmony, that the body remains balanced as a whole. Perhaps most remarkably, mitochondria communicate across generations. Their DNA, passed down maternally, carries not only the instructions for energy production, but also the marks of adaptations to past environments. Variations in mitochondrial DNA can influence metabolism, adaptation to climate, and susceptibility to disease. These variations are messages from the past, whispers of the environments in which our ancestors lived. Each mitochondrion, through its DNA, tells a story that extends beyond the present moment, linking us to the history of our species. This communicative role reshapes how we think about mitochondria. They are not passive organelles, but active participants in the decisions of life and death, growth and rest, survival and adaptation. Their signals guide the cell like a quiet conversation partner. The nucleus may hold the instructions, but mitochondria provide the commentary, the feedback that shapes how those instructions are carried out. Together, they form a dialogue, a constant back and forth that keeps the cell alive and responsive to its environment. It is soothing to imagine this dialogue as a gentle murmur within the cell. Signals move back and forth, molecules flowing like words, instructions exchanged in silence. Mitochondria whisper to the nucleus about energy levels, about stress, about the need for renewal or rest. The nucleus listens and responds, adjusting gene expression, producing proteins, altering the course of the cell's behavior. This is not a loud conversation, but a subtle one, unfolding continuously without pause, as seamless as breathing, as constant as the beat of the heart. Even as you rest, this dialogue continues within you. Your mitochondria are not only producing ATP, but also guiding your cells, sending signals that shape your health, your adaptation, your resilience. They are communicators as much as they are engines. Their voices woven into the fabric of your biology. Their communication is not heard in sound, but felt in balance, in the steady maintenance of life's processes, in the quiet assurance that your cells are guided by signals older than memory. And so, mitochondria remind us that life is not sustained by power alone, but also by communication. Energy without guidance would be chaos, but energy shaped by signals becomes life. Within you, countless mitochondria are speaking in their own language, regulating, guiding, maintaining harmony. They are not only the powerhouses of the cell, but also its messengers, its guardians, its quiet voices of balance. Their communication is ceaseless, and in that ceaselessness lies the secret of life's endurance. 14. Mitochondria and aging. Aging is one of the most universal experiences of life. From the smallest creatures to the most complex organisms, the passage of time leaves its mark. Cells divide, tissues repair, bodies grow, and eventually, these processes begin to slow, to falter, to wear down. While aging is shaped by many factors, mitochondria stand at the heart of this story. Their constant work, their endless production of energy, and their role in signaling and repair all tie them to the gradual unfolding of age. To look closely at mitochondria is to glimpse part of the mystery of why we grow older, why energy fades, and why time leaves its gentle imprint upon us all. Throughout life, mitochondria produce ATP, fueling the countless demands of the body. Yet in doing so, they also create byproducts, small molecules called reactive oxygen species, or ROS. These are generated when electrons leak from the electron transport chain and react with oxygen. In moderation, ROS are useful, acting as signals that help regulate growth and adaptation. But in excess, they can damage proteins, lipids, and DNA. Over years and decades, this damage accumulates, affecting not only the cell, but also the mitochondria themselves. Their DNA, lacking the same protective structures as nuclear DNA, mutations build, impairing mitochondrial function, reducing their efficiency, and leading to further production of ROS in a cycle that gradually contributes to aging. This is sometimes called the mitochondrial free radical theory of aging, a framework that suggests that wear and tear from reactive molecules plays a central role in the decline of cellular function over time. Though modern science sees aging as a far more complex process, with many contributing causes, mitochondria remain a key element. Their central role in energy production means that any decline in their efficiency resonates throughout the body. As mitochondria falter, cells receive less ATP, processes slow, and the tissues of the body begin to feel the weight of years. Muscles provide a clear example of this change. In youth, they are strong and resilient, fueled by abundant mitochondria, producing ample energy. With age, however, mitochondria decline in both number and function. Muscle fibers weaken, endurance fades, and recovery slows. This is why physical strength tends to diminish with time. The mitochondria, once abundant forges of energy, gradually lose their edge, and the body adjusts to this quieter rhythm. The brain, too, feels the effects of mitochondrial aging. Neurons are heavily reliant on mitochondria, requiring vast amounts of ATP to maintain their ion gradients and transmit signals. As mitochondria become less efficient, neurons struggle to keep up with their energy needs. Cognitive functions may slow, memory may weaken, and the brain becomes more vulnerable to diseases linked to mitochondrial dysfunction. Conditions such as Parkinson's and Alzheimer's have been associated with mitochondrial decline, further underscoring the connection between these organelles and the aging process. The heart, tirelessly beating from birth until the final moment of life, is another tissue deeply dependent on mitochondria. Cardiac cells contain an extraordinary density of these organelles, and their decline with age contributes to the weakening of the heart. While the heart continues to beat, its resilience fades, its ability to adapt to stress lessens, and the risks of disease increase. Mitochondrial health and cardiac health are bound together, and as mitochondria age, so too does the heart. Yet mitochondria are not merely passive victims of time. They are also active participants in regulating aging. Through their signals, they influence processes such as autophagy, the recycling of damaged cellular components, and apoptosis, the removal of cells that can no longer function properly. When these processes are balanced, aging proceeds gently, with tissues maintaining health for as long as possible. When the balance is lost, aging accelerates, and disease emerges. Mitochondria thus act as both markers of age and regulators of how gracefully or harshly it unfolds. There is also a paradox in their role. While mitochondria produce reactive oxygen species that can damage cells, these same molecules can trigger adaptive responses that strengthen defenses. In small amounts, ROS stimulate the cell to increase antioxidant production, to repair damage, to enhance resilience. This phenomenon, called hormesis, suggests that low levels of stress may actually promote health and longevity. Mitochondria, therefore, are not simply agents of decline, but also of adaptation. They walk a fine line between harm and help, between fragility and strength. This dual nature has inspired research into how mitochondrial function might be preserved or even enhanced with age. Exercise, for instance, is known to stimulate the production of new mitochondria, a process called mitochondrial biogenesis. By challenging the body, exercise encourages mitochondria to renew themselves, to multiply, and to maintain efficiency. In this way, the natural decline of aging can be slowed, though never entirely stopped. Nutrition, too, plays a role, with certain dietary patterns influencing mitochondrial health. While these insights are part of modern science, they are also reminders that mitochondria remain central not only to life, but to the pace at which it unfolds. Aging, when seen through the lens of mitochondria, becomes not just decline but also continuity. Though mitochondria falter, they continue to work, to provide energy, to support life until its final moment. Their decline is gradual, their signals persistent, their contributions unceasing. They are companions across the entire span of life, from the earliest divisions of embryonic cells to the final breaths of old age. Their journey is our journey, their fading glow a mirror of the passage of time itself. It is comforting to think of aging not as a sudden break, but as a gentle dimming, a gradual shift in rhythm. The mitochondria, once vibrant and efficient, slowly quiet, but their presence remains. Their work continues. They have carried us through growth, through strength, through maturity, and they remain with us in age, still sustaining, still supporting, still keeping the flame alive. They are not lost, only changed, adapting to the slow drift of years. As you reflect, imagine mitochondria as lanterns glowing within every cell. In youth, they shine brightly, their light strong and abundant. With time, the glow softens, flickers gently, yet still warms the space around it. The lanterns may dim, but they do not vanish. They continue to illuminate, even as the years lengthen. And in their light lies the story of aging. Not an end, but a transformation, a shift in balance, a quieting of the song of energy. Thus, mitochondria and aging are inseparable. They shape how time touches us, how energy flows through us, how resilience is maintained or lost. They are both the engines of youth and the companions of age, both the source of vitality and the markers of decline. To understand them is to glimpse part of the mystery of time itself, part of the gentle truth that life is not fixed, but always changing, always moving forward. And as you rest, your mitochondria continue their ceaseless work, their lanterns still glowing, their energy still flowing. They are with you in youth, in age, in every moment of your journey. In their steady hum lies the quiet rhythm of aging, a rhythm that carries you softly forward toward the unfolding of life's story, one breath, one beat, one gentle moment at a time. 15. Mitochondrial diseases. While mitochondria are most often celebrated as the powerhouses of the cell, providing the energy that fuels every moment of life, they also remind us of the fragility that lies within all living systems. For when mitochondria falter, the effects ripple outward, touching every part of the body. The diseases and conditions linked to mitochondrial dysfunction are as varied as the roles mitochondria play, and though many of these illnesses are rare, their stories reveal just how central mitochondria are to health. To speak of mitochondrial diseases is not to focus on fear, but to understand the deep dependence we have on these small organelles, and to appreciate the remarkable balance they maintain in sustaining life. Because mitochondria produce ATP, any disruption in their function results in an energy deficit. Cells that require large amounts of energy are especially vulnerable. These include the muscles, which contract constantly, the brain, which demands energy to transmit signals, the heart, which beats without pause, and the eyes, which process light and movement without rest. When mitochondria fail to meet the demands of these tissues, the results can be profound. Muscle weakness, neurological difficulties, heart problems, and vision loss are among the symptoms that can appear when mitochondria cannot keep pace with the needs of the body. Some mitochondrial diseases arise from inherited mutations in mitochondrial DNA. Because mtDNA is passed maternally, such conditions are transmitted through the maternal line. If a mother carries mutations in her mitochondrial DNA, she may pass them on to her children, though the severity can vary depending on how many of her mitochondria carry the mutation. This variability arises because each cell contains many mitochondria, and not all of them may be affected. The mixture of normal and mutated mitochondria in a cell is called heteroplasmy, and it determines whether symptoms are mild, severe, or even absent. Other mitochondrial disorders come from mutations in nuclear DNA that affect proteins used by the mitochondria. Since mitochondria rely on both their own genes and those encoded in the nucleus, problems can arise from either source. These mutations may interfere with the electron transport chain, with ATP synthase, or with the enzymes of the citric acid cycle. No matter where the disruption occurs, the result is the same: a shortage of ATP, a slowing of processes, a weakening of tissues. The diversity of mitochondrial diseases makes them difficult to classify. Some affect only one organ, while others spread their impact across multiple systems. Mitochondrial myopathies, for example, primarily affect the muscles, leading to weakness, exercise, intolerance, and fatigue. Lee syndrome, a severe neurological disorder, arises in infancy or childhood and leads to progressive loss of mental and motor abilities. Other conditions affect the eyes, such as Leber's hereditary optic neuropathy, which causes vision loss due to damage in the optic nerve. Each of these diseases tells the same underlying story. When mitochondria cannot provide enough energy, the tissues most dependent on them falter. Beyond rare genetic conditions, mitochondrial dysfunction is also implicated in more common diseases of aging and chronic illness. Research has linked mitochondrial decline to Parkinson's disease, Alzheimer's disease, diabetes, and cardiovascular disorders. In these cases, the mitochondria may not be the sole cause, but their inefficiency contributes to the progression of the illness. The neurons of the brain, the cells of the pancreas, the fibers of the heart all depend heavily on mitochondria, and when these organelles are stressed, the tissues show signs of strain. Thus, mitochondrial health is not only a matter of rare inherited diseases, but also a factor in many conditions that affect millions worldwide. It is striking that symptoms of mitochondrial dysfunction often resemble the signs of aging. Fatigue, muscle weakness, memory loss, and reduced resilience all appear both in mitochondrial diseases and in natural aging. This overlap reinforces the idea that mitochondria are at the core of vitality, and that their decline is closely tied to the passage of time. Whether through inherited mutations or gradual wear, the story is the same. As mitochondria weaken, so too does the energy of life. Despite the challenges posed by mitochondrial diseases, there is a resilience in the body. Cells sometimes compensate by increasing the number of mitochondria, attempting to make up for the inefficiency of the faulty ones. This adaptation does not always solve the problem, but it shows the determination of life to endure. Researchers, too, continue to explore ways of supporting mitochondrial function, from therapies that aim to bypass defective pathways to approaches that enhance the body's natural defenses against oxidative stress. Though these conditions remain difficult to treat, the study of them has deepened our understanding of mitochondria and their role in health. For those who live with mitochondrial disorders, the experience is one of adjustment. Energy may be limited, and the body may respond unpredictably. Yet within these challenges lies a profound truth. Even when mitochondria falter, life continues. The cells adapt, the body persists, and individuals find ways to live within the rhythm their mitochondria provide. The resilience of life is not only in its abundance of energy, but also in its ability to endure when energy is scarce. When reflecting on mitochondrial diseases, it is possible to see them not only as medical conditions, but also as reminders of our deep dependence on balance. They show us how finely tuned the processes of life are, how even the smallest organelles play essential roles, and how the harmony of the body can be disrupted when one part weakens. They remind us of the interconnectedness of systems, of the way energy flows through us, and of the fragility that lies beneath our strength. It can be soothing to imagine mitochondria as lanterns within the body, most glowing brightly, some flickering more faintly. In health, the lanterns burn steadily, illuminating every tissue, every cell. In mitochondrial disease, some lanterns dim, casting shadows in certain places. Yet even in the shadows, other lanterns continue to shine, sustaining life, carrying forward the flame. The body is not lost, only altered, adapting to the pattern of light it is given. And so, the story of mitochondrial diseases is not merely one of decline, but also of endurance. It is the story of how cells cope with scarcity, how tissues adjust to weakness, how life finds ways to continue even when its engines struggle. It is a reminder that mitochondria are central not only to health but to the vulnerabilities of life, that in their strength we find vitality, and in their weakness we glimpse the quiet fragility that makes us human. As you reflect on this, let the thought settle gently. Within you, countless mitochondria are still at work, sustaining your life in every moment. Some may falter, as they do in disease, but the vast majority continue their ceaseless task. They are your companions, your silent workers, your faithful partners, and even when they struggle, they remain part of you, still glowing, still carrying forward the quiet rhythm of life. 16. Exercise and renewal. Among the many influences that shape the health of mitochondria, few are as powerful as movement. Exercise, whether it is the quiet rhythm of walking, the steady effort of swimming, or the focused intensity of running, brings renewal to the body. It awakens processes deep within the cell, stirring mitochondria to grow, to multiply, and to become more efficient. Far from wearing the body down, physical activity invites it to adapt, to strengthen, and to sustain itself with greater vitality. When we speak of exercise, we often think of muscles, of strength or endurance, but at the most intimate level, exercise is a conversation between movement and mitochondria, a dialogue that shapes the very flow of energy within. When muscles contract repeatedly during activity, their demand for energy rises dramatically. A resting muscle may rely on a modest trickle of ATP, but during exercise, the demand can surge many times higher. To meet this, mitochondria increase their output, drawing upon oxygen and nutrients to produce more ATP through oxidative phosphorylation. If the activity continues, the muscle sends signals that more mitochondria are needed. These signals activate pathways that promote mitochondrial biogenesis, the process by which new mitochondria are formed within the cell. Over time, repeated bouts of exercise lead to a greater number of mitochondria, each ready to share in the task of supplying energy. This adaptation explains why endurance improves with training. The body is not only learning to move, but also reshaping its very cells to support sustained effort. The renewal of mitochondria through exercise is not only about numbers. Existing mitochondria also become more efficient. They fine-tune their electron transport chains, improve their capacity to manage reactive oxygen species, and strengthen their ability to use oxygen. In essence, they become better at their work. This improvement is not confined to muscles alone. The benefits extend to the heart, which pumps more steadily and efficiently, to the lungs, which deliver oxygen more effectively, and to the brain, which receives greater blood flow and enhanced support from its own mitochondria. In this way, exercise is a full-body renewal, a stimulus that echoes through every tissue. One of the remarkable aspects of exercise-induced renewal is its effect on aging. As we saw before, mitochondria naturally decline with age, becoming fewer in number and less efficient. Exercise slows this decline. It stimulates the pathways that keep mitochondria healthy, encouraging their growth even in older adults. Studies have shown that regular physical activity preserves mitochondrial function in aging muscles, allowing older individuals to maintain strength, endurance, and vitality longer than those who remain sedentary. In this sense, exercise is not only a source of renewal in youth but also a guardian in age, preserving mitochondrial health across the span of life. The influence of exercise on mitochondria also touches the realm of disease. In conditions linked to mitochondrial dysfunction, physical activity can act as a therapy, stimulating the creation of healthier mitochondria and enhancing the resilience of tissues. While it may not cure such conditions, it often alleviates symptoms, improves quality of life, and strengthens the body's ability to adapt. Even in individuals without illness, exercise fortifies mitochondrial networks, creating a reserve of energy capacity that supports health in countless ways. The connection between exercise and mitochondria also highlights the body's remarkable adaptability. Muscles trained for endurance grow rich in mitochondria, enabling them to sustain long, steady efforts. Muscles trained for strength adapt differently, focusing on power but still drawing upon mitochondria for recovery and repair. No matter the form, movement calls upon mitochondria, and mitochondria respond, shaping themselves to the rhythm of activity. It is a partnership that reflects the deep connection between motion and life itself. At a molecular level, this renewal is guided by signaling pathways that act like messengers within the cell. One such pathway involves a protein called PGC1, often described as a master regulator of mitochondrial biogenesis. When activated by exercise, PGC1 turns on genes that encourage the growth of new mitochondria and the strengthening of existing ones. This response is part of the elegant language of biology, a way the cell translates the experience of movement into lasting change. It is as though the cell takes note of the demand, remembers it, and prepares better for the next time. Beyond the physical, exercise also influences the mitochondria of the brain. Movement stimulates the release of molecules that support neurons, encourage the growth of new connections, and enhance mitochondrial function. This is one reason why physical activity is linked to improved mood, sharper memory, and resilience against cognitive decline. The mitochondria of the brain, like those of the muscles, thrive under the influence of activity, finding renewal in the flow of oxygen and energy. It is soothing to imagine mitochondria responding to exercise like tiny lanterns within the cell. As movement begins, the lanterns glow brighter, producing more energy, illuminating the cell from within. With continued practice, more lanterns appear, each adding to the light, each strengthening the glow. Over time, the tissue becomes not only more energetic, but also more resilient, better able to withstand the challenges of life. The lanterns burn not with exhaustion but with renewal, reflecting the paradox that effort, when balanced, leads not to depletion, but to strength. The renewal brought by exercise is not abrupt or dramatic. It is gradual, unfolding over days and weeks, as mitochondria multiply and adapt. Each step, each breath, each movement contributes to this transformation, shaping the body quietly, invisibly, from within. The result is not only improved performance but also a deeper sense of vitality, a resilience that supports life in all its aspects. Even gentle forms of activity, like walking or stretching, carry this benefit. For every call to movement is also a call to the mitochondria, an invitation to renew. In reflecting on this, one can see exercise not merely as effort but as conversation. It is a dialogue between the outer world of movement and the inner world of mitochondria. Each time the body moves, mitochondria listen, respond, and adapt. Each time they renew themselves, they prepare the body for more, creating a cycle of interaction that sustains vitality. This cycle is one of the quiet truths of life, that through motion, energy is not only spent but replenished, not only consumed but restored, and so, exercise and renewal are inseparable. The mitochondria respond to movement with growth, efficiency, and resilience. They adapt not only to the demands of today but also to the needs of tomorrow, carrying forward the benefits of each step, each breath, each effort. They are partners in this renewal, lanterns glowing brighter with every movement, sustaining the body through youth, through age, through the passage of time. As you rest now, it is calming to know that even gentle movements earlier in the day have left their mark, that your mitochondria have listened and responded. They continue to renew themselves quietly, preparing you for tomorrow, for the next step, for the next breath. Their renewal is ceaseless, their adaptation constant, their presence enduring. In their glow lies the quiet assurance that life, through movement, finds strength not in depletion, but in renewal. 17 Mitochondria and stress response. Life, even in its calmest forms, is never free from stress. Stress is not only the strain of emotion or the challenges of daily existence. It is also the countless small disturbances that cells encounter every moment. Changes in temperature, fluctuations in nutrients, sudden surges in energy demand, exposure to toxins, or the simple wear and tear of living. All of these are forms of stress. Mitochondria, as the powerhouses and communicators of the cell, are at the very center of how the body responds. They do not merely endure stress. They sense it, interpret it, and send signals that shape how the cell will adapt. Their responses can guide a cell toward resilience and recovery, or, in times of overwhelming strain, toward the decision to step aside and make way for new life. The most immediate way mitochondria respond to stress is through their ability to regulate energy. When a cell suddenly requires more ATP, mitochondria shift into higher activity, accelerating the electron transport chain, drawing on more oxygen, and releasing more energy. This responsiveness allows cells to adapt quickly to demands such as sudden movement, intense thought, or unexpected injury. The mitochondria adjust without hesitation, ensuring that the flow of ATP matches the needs of the moment. In doing so, they serve as the cell's first responders, quietly stabilizing the energy supply. But stress is not always so simple. Some stresses threaten the very integrity of the cell. When toxins or extreme conditions disrupt the electron transport chain, mitochondria may release reactive oxygen species in higher amounts. These molecules, while potentially damaging, also act as signals, alerting the nucleus that conditions are unfavorable. The cell then activates pathways to repair proteins, increase antioxidant defenses, and restore balance. In this way, mitochondria use stress as a form of communication, sending warning signals that allow the cell to adapt. Far from being destructive, this controlled release of reactive molecules can strengthen the cell's resilience, preparing it for future challenges. Mitochondria also respond to stress through changes in their shape and organization. They are not fixed structures but dynamic, constantly undergoing fission and fusion. Fusion allows mitochondria to join together, sharing contents and compensating for localized damage. Fission, by contrast, separates damaged portions, isolating them so that they can be removed through autophagy, the process of recycling cellular components. This dynamic behavior ensures that mitochondria remain healthy, replacing worn parts, removing broken fragments, and renewing their function. During times of stress, the balance between fusion and fission shifts, reflecting the cell's needs. This flexibility is one of the ways mitochondria maintain their endurance in the face of constant challenges. When stress becomes too great, however, mitochondria can initiate apoptosis, the programmed death of the cell. They release proteins that activate a cascade of signals, leading to the dismantling of the cell in a controlled manner. This is not an act of failure, but of protection. By removing cells that are too damaged to recover, mitochondria safeguard the organism as a whole. The decision to initiate apoptosis reflects the mitochondria's role as guardians, weighing the balance between survival and sacrifice. In this sense, they embody a wisdom of life itself, the ability to recognize when persistence is beneficial and when letting go is the better path. Stress also influences mitochondria beyond the level of individual cells. When tissues experience strain, mitochondria release signals that attract immune cells, coordinate inflammation, and promote healing. This response can be beneficial, allowing the body to repair itself after injury. But if the signals persist for too long, they can contribute to chronic inflammation, a condition linked to aging and many diseases. Thus, mitochondrial responses to stress are both protective and delicate, requiring balance to avoid tipping into harm. The stress response also connects mitochondria to the body's broader systems. Hormones released during times of emotional or physical stress, such as cortisol and adrenaline, influence mitochondrial activity. These signals prepare the body to respond, increasing energy production, heightening alertness, and mobilizing resources. At the same time, mitochondria send feedback signals that influence how long and how intensely the stress response continues. They help the body know when to remain vigilant and when to return to calm. In this way, mitochondria are participants in the dialogue between mind and body, shaping how stress is experienced and resolved. Interestingly, not all stress is harmful. Just as the body strengthens with moderate exercise, cells strengthen with mild stress. When mitochondria experience low levels of strain, they adapt by becoming more efficient, producing more antioxidants, and enhancing their resilience. This principle, called hormesis, shows that stress in moderation can be a source of renewal. It is not the absence of stress, but the right balance of stress that fosters health. Mitochondria are central to this process, translating small challenges into lasting strength. To picture mitochondria under stress, one might imagine a fleet of lanterns flickering in the wind. Some burn more brightly to compensate, others fuse together to shield their light, and a few extinguish themselves so the others may endure. The lanterns communicate, adapt, and adjust until balance is restored. Even in the presence of strain, the light continues, not as a steady blaze, but as a resilient glow that persists against the breeze. This imagery reflects the quiet persistence of mitochondria, their ability to endure and respond without ceasing. Even now, as you breathe calmly, your mitochondria are engaged in countless small stress responses. They adjust to fluctuations in nutrients, to changes in temperature, to signals from hormones, to the ordinary wear of living. They respond silently, without demand for attention, keeping balance within each cell. They are guardians of harmony, sensing strain, releasing signals, repairing damage, and choosing wisely when to persist and when to let go. And so, mitochondria remind us that stress is not only a burden but also a teacher. It is through stress that they grow stronger, that they signal renewal, that they maintain balance. They reveal that resilience is not the absence of strain, but the ability to respond to it with wisdom and adaptability. Within you, mitochondria embody this lesson, ceaselessly responding, ceaselessly adapting, ceaselessly keeping the rhythm of life steady in the face of change. Their response to stress is thus a reminder of life's quiet strength. Like lanterns glowing in the wind, They do not resist the storm with force, but with adaptation, with communication, with balance. And in their persistence lies the secret of survival, the assurance that even in times of strain, the light of life continues to glow, steady, enduring, unbroken. 18. The link between mitochondria and immunity. The immune system is often described as the body's defense network, a vigilant guardian that stands ready to protect against invading microbes, toxins, and disruptions. Yet beneath the visible layers of this defense lies a deeper foundation, one that depends on the quiet work of mitochondria. Though we may think of immunity as the realm of white blood cells, antibodies, and signaling molecules, mitochondria are woven into this fabric, shaping how the immune system responds, adapts, and remembers. They are not separate from immunity, but partners in its function, providing both energy and guidance. To explore this link is to see mitochondria not only as engines of life, but also as silent protectors, sustaining resilience against the challenges that come from beyond and within. The most obvious contribution mitochondria make to immunity is energy. Every action of the immune system requires ATP, from the movement of white blood cells to the synthesis of antibodies, from the signaling between cells to the destruction of pathogens. When a macrophage engulfs a bacterium, when a T cell recognizes an infected cell, when a B cell releases antibodies, all of these tasks are fueled by mitochondrial energy. Without mitochondria, immune cells would lack the strength to carry out their roles, and the defense of the body would falter. Yet mitochondria do more than provide fuel. They also act as regulators, shaping how immune cells behave. When immune cells are activated, they often change their metabolism, switching from one source of energy to another. For example, certain T cells shift from relying on oxidative phosphorylation to using glycolysis, a process that allows them to generate energy quickly for rapid responses. Mitochondria coordinate these shifts, adjusting their activity and signaling pathways to match the needs of the immune system. In this way, they are not passive suppliers of ATP but active guides, helping immune cells choose the right strategies for each challenge. One of the most fascinating aspects of this partnership is the way mitochondria serve as sentinels of danger. When cells are damaged, mitochondria can release fragments of their own DNA into the cytoplasm or bloodstream. Because mitochondrial DNA resembles bacterial DNA, the immune system recognizes it as a potential alarm signal. This triggers pathways that alert immune cells to the presence of damage or infection. Thus, mitochondria themselves can act as danger signals, summoning the body's defenses to sites of stress or injury. It is as if the mitochondria carry with them not only the energy of life but also the language of warning, able to call for help when balance is lost. Mitochondria are also involved in the production of molecules that guide inflammation, the process by which the immune system responds to threats. Through their metabolic activities, they help generate signals such as cytokines, which coordinate the activity of immune cells. Inflammation is a double-edged sword, necessary for defense and healing, yet potentially harmful if uncontrolled. Mitochondria, by regulating energy and signaling, help keep this process balanced, allowing the body to fight infection while minimizing damage to its own tissues. In the context of viral infections, mitochondria play a particularly intriguing role. Within their membranes reside proteins that detect viral RNA, initiating pathways that lead to the production of interferons, powerful molecules that help cells resist viral replication. This mitochondrial signaling is one of the first lines of defense against viruses, demonstrating again that mitochondria are not passive but active participants in immunity. They are guardians within, ready to recognize intruders and to send alerts that mobilize the broader immune system. The connection between mitochondria and immunity also extends to memory. One of the hallmarks of the immune system is its ability to remember past encounters, to respond more quickly and effectively to pathogens it has seen before. Recent research suggests that mitochondria contribute to this memory by influencing how long-lived certain immune cells become and how they maintain their readiness. The health and function of mitochondria in these cells determine whether the memory endures, linking mitochondrial resilience to the body's ability to recall and respond across a lifetime. There are also times when the link between mitochondria and immunity becomes strained. In autoimmune diseases, where the immune system mistakenly attacks the body's own tissues, mitochondrial signals may contribute to the confusion. Fragments of mitochondrial DNA released during stress can be misinterpreted as foreign, fueling inappropriate immune responses. Similarly, when mitochondria are damaged by chronic stress, toxins, or aging, their altered signals can disrupt immune balance, contributing to conditions of inflammation and disease. These examples reveal how closely immunity depends on mitochondrial harmony, and how quickly imbalance can ripple outward into disorder. To imagine this partnership, one might picture mitochondria as lanterns illuminating a vast city at night. The immune system is the city's guard, moving through the streets, scanning for intruders, ready to act. The lanterns not only light the way, but also send signals flickering when danger is near, glowing steadily when balance is kept. Without the lanterns, the guard would be blind and powerless. With them, the guard is guided, strengthened, and sustained. This imagery captures the quiet yet profound role mitochondria play in immunity, their glow both energizing and directing the defense of life. Even now, as you rest, your mitochondria are part of this defense. They are fueling the constant vigilance of immune cells, sustaining the quiet patrols that continue even in sleep. They are producing the signals that maintain readiness, that balance inflammation, that guide the immune system toward harmony. Their role is ceaseless, silent, and vital, ensuring that the defenses of the body remain strong without your ever having to think of them. And so, mitochondria are more than powerhouses. They are guardians, guides, and communicators, bound intimately to the immune system. They sustain its strength, direct its responses, and contribute to its memory. In their signals lie both the power to defend and the need for balance, reminding us that immunity, like energy, is a matter of harmony. Their link to immunity reveals once again that mitochondria are central not only to life but to the resilience of life against challenge. As you let your thoughts settle, it is soothing to remember that within you, mitochondria are working not only to keep your heart beating and your mind awake, but also to keep your body safe. They are lanterns in the night, glowing quietly, guiding the immune system, sustaining the balance that allows you to rest in peace. Their partnership with immunity is one of life's quiet wonders, a reminder that even the smallest organelles can hold the greatest responsibilities, carrying forward the strength of the body against all that might disturb its calm. 19 mitochondria in different organisms. Mitochondria are present in nearly all eukaryotic life, from the simplest single-celled organisms to the most complex animals. Their presence is universal, yet their forms and functions reveal fascinating variations that reflect the diversity of life itself. To study mitochondria across different organisms is to see how a single ancient partnership between a host cell and a bacterium blossomed into countless adaptations, each shaped by the environment, by necessity, and by the rhythm of survival. Though they all share the same core role of producing energy through oxidative phosphorylation, mitochondria have taken on specialized tasks in different species, reminding us that life's unity is always paired with variety. In single-celled eukaryotes, mitochondria often appear in their most versatile forms. Protists, for example, show a wide range of mitochondrial adaptations. Some live in oxygen-rich environments, where mitochondria perform the classic role of producing ATP through the electron transport chain. Others, however, inhabit environments where oxygen is scarce or absent. In these cases, Mitochondria have evolved into modified structures called hydrogenosomes or mitosomes. Hydrogenosomes generate ATP without oxygen, releasing hydrogen gas as a byproduct, while mitosomes have lost most of their energy-producing capacity, but still retain essential roles in metabolism. These variations show that mitochondria are not rigid in their design. They adapt to the conditions of their environment, finding ways to sustain life even in places where oxygen is absent. In fungi, mitochondria take on roles that support the unique lifestyles of these organisms. Yeast cells, for instance, are capable of both fermentation and respiration. When oxygen is abundant, their mitochondria produce ATP efficiently through oxidative phosphorylation. When oxygen is scarce, they shift to fermentation, producing energy less efficiently, but still enough to survive. This flexibility has made yeast invaluable in human culture, from bread making to brewing, but it also reflects the adaptability of their mitochondria, able to switch between strategies as conditions demand. Plants, too, contain mitochondria, though they often go unnoticed because of the presence of chloroplasts. Chloroplasts capture sunlight and produce energy-rich molecules through photosynthesis. Yet mitochondria remain essential. During the night, when photosynthesis ceases, mitochondria take over the primary role of producing ATP, ensuring that cells remain alive and active even without sunlight. In fact, mitochondria and chloroplasts often work together, exchanging metabolites and balancing the energy needs of the plant. This partnership reflects a broader theme. Mitochondria are not isolated engines but collaborators, adapting their roles to fit the larger network of the cell. In animals, mitochondria are most abundant in tissues with high energy demands, such as muscles, nerves, and sensory organs. Yet the specific ways mitochondria adapt differ among species. Birds, for example, have mitochondria that are especially efficient at producing ATP, supporting the high metabolic cost of flight. The demands of sustained flight, with its rapid wing beats and intense oxygen consumption, require mitochondria that are finely tuned to maintain energy flow without faltering. Similarly, in diving mammals such as whales and seals, mitochondria adapt to function efficiently during extended periods without oxygen, ensuring that muscles remain supplied with ATP even while underwater. These adaptations show how mitochondria reflect the lifestyles of their hosts, sculpting their capacities to match the needs of survival. Insects present another fascinating example. Their mitochondria support the rapid muscle contractions required for flight, generating ATP at extraordinary rates. Yet insects also reveal how mitochondria can contribute to more than energy. In some species, mitochondria play roles in development, reproduction, and even in determining sex through their interactions with symbiotic bacteria. These specialized functions highlight the versatility of mitochondria, showing that their contributions are not limited to metabolism alone, but extend to the broader patterns of life. Even within humans, mitochondria show variation that reflects adaptation to different environments. Populations living in colder climates often have mitochondrial variants that favor heat production, while those in warmer regions may have variants that emphasize efficiency. These subtle differences are inherited through mitochondrial DNA, passed down through maternal lines, and they reflect the way mitochondria continue to shape our adaptation to the world. In this sense, mitochondria are not only remnants of ancient history, but also active participants in ongoing evolution. The diversity of mitochondria across organisms also raises questions about the unity of life. Despite their differences, all mitochondria share common features: their double membranes, their circular DNA, their ribosomes, and their core role in energy production. This unity points back to their shared origin, the moment when a single bacterium entered into symbiosis with a host cell more than 1 billion years ago. From that moment, countless lineages diverged, each carrying mitochondria within them, each shaping those organelles to their own needs. The mitochondria of a yeast cell, of a plant, of a bird, of a human, all are variations on the same theme, threads woven from the same ancient fabric. To imagine mitochondria in different organisms is to see a gallery of adaptations. In the deep ocean, protists with hydrogenosomes thrive without oxygen, their mitochondria altered to fit the darkness. In forests, plants rely on both chloroplasts and mitochondria, the two organelles working side by side to sustain growth and survival. In the skies, birds carry mitochondria fine-tuned for flight, while in the seas, whales carry mitochondria adapted to the stillness of long dives. Across all these examples, mitochondria remain central, their presence a constant, even as their forms vary. This universality is humbling. It reminds us that the mitochondria within our own cells are not unique to us but part of a vast lineage that stretches across the tree of life. They link us to yeast in a loaf of bread, to trees swaying in the wind, to birds in flight, to creatures of the deep. They are the common thread of eukaryotic existence, the shared inheritance of all beings that carry nuclei and organelles. In their unity and diversity, mitochondria embody both the sameness and the difference of life, showing that while the world is filled with variation, it is also bound by common origins. Even now, as you breathe gently, mitochondria in your own cells are carrying out the same processes as those in a plant leaf, a yeast cell, or a bird's wing. They are producing ATP through the flow of electrons, maintaining balance, adapting to the moment. Their glow is the same glow that sustains countless other organisms, a light shared across the living world. In this way, mitochondria remind us that we are never isolated but always part of a greater tapestry, woven together by the shared rhythms of energy and life. And so, mitochondria in different organisms tell a story of diversity and connection. They reveal how life adapts to its environments, how a single ancient event blossomed into a multitude of forms, how unity and variety are woven together. They remind us that the same organelles sustaining our own lives are also sustaining the lives of countless others, each adapted to its own path, each carrying forward the legacy of that first partnership. In their diversity, mitochondria reveal the richness of life. In their unity, they reveal its shared foundation. 20. The centrality of life. To speak of mitochondria is, in many ways, to speak of life itself. These small organelles, nestled deep within the folds of cells, are not merely helpers or accessories, but the very foundation upon which complex life rests. They are at once ancient and present, independent and integrated, fragile and powerful. From the earliest whispers of evolution to the beating of your own heart, mitochondria stand at the center, shaping the rhythms of existence. To reflect on their centrality is to glimpse not only the mechanics of biology, but also the quiet truth that life is built upon connection, upon balance, and upon the ceaseless flow of energy. Energy is the thread that weaves through all living things. Without it, cells would be still, silent, unable to divide, unable to repair, unable to think, or move, or dream. Mitochondria provide this energy in a form that can be spent and renewed, a currency of ATP that powers every action. They are the forges in which the potential stored in food is transformed into motion, into thought, into warmth, into life itself. The centrality of mitochondria lies in this transformation, in their ability to make energy available in abundance, to sustain not just survival, but complexity, not just endurance, but flourishing. Consider the diversity of processes that depend on ATP. Muscles contracting in steady rhythm, neurons firing to create the experience of memory, enzymes folding proteins into precise shapes, immune cells patrolling for threats. Each of these draws upon the energy minted by mitochondria. The organelles do not choose where their ATP is spent, yet they are present in every action, woven invisibly into the fabric of being. Their centrality is not loud or obvious. It is quiet, constant, and universal, like the hum of a river flowing beneath the surface of a landscape. The centrality of mitochondria also lies in their role as communicators. They do not simply generate energy in isolation. They inform the cell of its condition, sending signals that guide growth, repair, and renewal. They regulate cell death when damage is too great, ensuring that the integrity of the organism is preserved. They shape immunity, sounding alarms when pathogens or damage threaten balance. They adapt to stress, adjusting their function, renewing themselves, and maintaining resilience. Their central role is not mechanical but guiding, not only fueling the cell but steering its choices, shaping the path it follows. Their importance extends beyond individual health to the story of evolution itself. The union that gave rise to mitochondria marked a turning point in the history of Earth, allowing cells to grow more complex, to harbor more DNA, to form multicellular organisms. From this partnership flowed the diversity of plants, animals, fungi, and countless other beings. Without mitochondria, life might have remained forever simple, confined to the scale of bacteria, never branching into the rich tapestry of ecosystems we see today. Their centrality is not only biological but historical, for they opened the path to all that has followed. Yet mitochondria are not only central to the story of life, they are central to the story of us. Passed down through maternal lines, they connect each person to their ancestors, unbroken through time. The mitochondria in your cells carry DNA that has journeyed across generations, through countless lives, across thousands of years. They are not only engines of your present life, but also carriers of memory, genetic whispers from the past. Their centrality is both functional and ancestral, linking each individual to the vast continuum of human history. Mitochondria also remind us of the delicate balance of life. Their production of energy is efficient, but it is never without risk. Reactive oxygen species, the byproducts of their activity, can damage cells if left unchecked. Yet in moderation, these same molecules serve as signals, strengthening defenses, guiding adaptation. This balance between help and harm, between creation and destruction, lies at the heart of life. Mitochondria embody this balance, showing us that centrality is not only about power but also about restraint, not only about abundance but also about harmony. It is soothing to imagine mitochondria as countless lanterns glowing within every cell. Their light is invisible, yet it sustains all that you are. Each lantern contributes to the whole, illuminating tissues, fueling thoughts, guiding actions. Some glow more brightly in muscles, others in neurons, others in the heart. Together, they form a constellation of light within you, a quiet galaxy of energy and life. Their centrality lies in this constellation, for without it, the body would be dark and still. With it, the body is vibrant, aware. Even in rest, mitochondria continue their ceaseless work. As you breathe gently, oxygen enters your cells, reaching the mitochondria, where it accepts electrons and becomes water. As you digest food, nutrients enter your blood, feeding the cycles of the mitochondria, producing ATP. You do not direct this, you do not feel it, yet it is happening in every moment. The centrality of mitochondria is quiet because it is constant. They do not demand attention. They simply continue, sustaining you without pause, without rest, from the first moment of life until the last. To reflect on mitochondria is to reflect on the unity of all living things. The same processes unfolding in your cells unfold in the cells of plants, of animals, of fungi, of countless other organisms. The same electron transport chain, the same ATP synthase, the same cycles of energy are repeated again and again across the living world. This universality reminds us that we are not separate but connected, bound by the same organelles, the same rhythms, the same flows of energy. The centrality of mitochondria is not confined to the human body. It is shared across the earth, a common thread in the tapestry of life. And so, mitochondria stand as symbols of both individuality and unity, of both past and present, of both fragility and strength. They are small, unseen, often overlooked, yet they are indispensable. They fuel every breath, every thought, every moment of existence. They connect us to our ancestors, to other species, to the earliest days of evolution. They embody the balance of creation and destruction, of energy and communication, of life and its passage through time. As you drift into calm reflection, let the thought settle that within you lies this central force, this quiet flame, this ceaseless hum of energy. It is the same flame that has burned through billions of years, passed from cell to cell, from generation to generation, from ancestor to descendant. It is the same hum that sustains the forests, the oceans, the skies. It is the rhythm of mitochondria, the centrality of life. And as you rest, their lanterns continue to glow, softly, steadily, carrying you forward in the quiet assurance that life, sustained by their energy, continues without pause.